Hello, everybody. Welcome back to Biochemistry. We have completed 7 days of sessions of biochemistry, molecular biology, and genetics, and we have 4 more days to go. Just 4 more days. Oh man. I'm just getting to know everybody, and now just 4 more days. Oh man. But in any case, I'm just so glad you're back on board because man, these next 4 days are really high yield stuff. You're going to see so much integration with pathology and pharm coming up here. So I'm glad you're on board, including tonight. Now, what we're going to do is you're going and we have tell the other faculty that like Dr. Barone and Dr. Harris that you guys are a good group to work with, fantastic group to work with, and so I'm so glad that that I've been a chance to interact with you doing these biochemistry reviews. All right, so where we're at is we're in chapter 10. We're going to finish off chapter 10 on vitamins with talking about one more vitamin. We talked about all the water-soluble vitamins, what you need to know about them in a nutshell. We talked about the 3 other fat-soluble vitamins and one more to talk about. And then after we're done talking about vitamin E, then there'll be there'll be 2 vignettes coming your way in front of you here, and we'll simulate the boards, how do the vignettes and make selections, and then we'll explain the results here. Okay, so the last vitamin that I wanted to talk about here is vitamin E. Now, vitamin E, there are about 7 different kinds of vitamin E in nature, and they're all called tocopherol. They have various potencies and so on, but they're all called tocopherol, alpha tocopherol, beta tocopherol. But in any case, what you need to know about them is what's the function of vitamin E. When you think vitamin A, D, and K, those were talked about yesterday. Blood clotting with vitamin K. Vitamin A has to do with vision or as acting as hormones, causing cells to grow. And vitamin D, you think calcium issues. But here now, vitamin E. When you think vitamin E, you think antioxidants. And so the idea is, that's why you see mostly a blank figure here on page 157, because on this figure is where we're going to be talking about free radicals. Why do you need antioxidants in the first place? And to understand why you need antioxidants, we got to talk about free radicals. And that's what's coming up on this particular page, a review of free radicals and why you need antioxidants. Now, just a couple more comments about vitamin E before we talk about free radicals. Without vitamin E deficiencies give rise to hemolysis, neurological problems, and retinitis, and so on. Now, vitamin E is deficiencies can give problems. On the other end of the spectrum, vitamin E overdose or vitamin E toxicity is virtually non-existent. So vitamin E is the only fat-soluble vitamin. In large amounts, excess amounts really doesn't cause any problems. The only time vitamin E in excess might cause a problem is right here. High doses of vitamin E can cause some people to hemorrhage when they're given warfarin. And the reason for that is if you were to look at the structure of vitamin E side by side with the structure of vitamin K, they look somewhat similar. And so in high doses of vitamin E, it can act as an antagonist of vitamin K action. And therefore, that's why a person might have a tendency to bleed, extra bleed. But we're not talking about bleeding to death, nothing like that. But simply the high doses of vitamin E in some patients can cause a little bit of bleeding problems because vitamin E can act as an antagonist of vitamin K action because it looks somewhat similar to vitamin K. No one expects you to know the structure of vitamin E, but it looks somewhat like vitamin K. Okay, now what we're going to talk about now is a review of free radicals. And you must understand free radicals because it is free radicals in the human body cause so many problems here. Now, when we think free radicals, we think oxygen. Now let me say something about oxygen. I got a lot to say about oxygen today, and you hear a lot about oxygen in physiology. But let me say something about oxygen. That oxygen is deadly for human beings. Yes, it's required for life, but it's also deadly for human beings. And the reason is because oxygen, I know students hate the term. I know students hate the term, but oxygen is the most oxidized form of the atom oxygen. Here we have O2, and that O2 oxygen atom is the most oxidized form of the atom oxygen. Now, what does that mean, the most oxidized form? That means that oxygen is an oxidant, oxidizing agent. It's looking for electrons. It's an electron acceptor. It's always looking for electrons. Now, how do you know oxygen is always looking for electrons? It's easy for me to say that, but how do you know that? And to illustrate this, if I took an iron ingot and put it on a table in front of me right here and come back next week, what would that iron ingot look like? Well, there'd be a coating of rust on the surface. Why? Because oxygen from the atmosphere comes down, takes the electrons from the iron and the iron ore, and thereby forming iron oxide, because oxygen takes electrons from the iron, because oxygen is always looking for electrons. And why does it want electrons? Because it wants to form the oxygen atom in water. And the oxygen atom in water is the most reduced form of the atom of oxygen. In other words, it finds the electrons that it's looking for. Now, let me stress here, oxidation reduction, the basis of human life or any life. Oxidation reduction always means electrons, not protons. When you think oxidation reduction, you only think electrons. Protons may accompany the electrons, but they don't have to. So the idea is when oxygen is looking for electrons to form the oxygen atom in water, how many electrons is it looking for? Let me see your answers here. How many electrons is oxygen looking for to form the harmless water? Not two. Good try. Lily, there it is. Lily got it, and the answer, and Vincent, and so on. And it's looking for 4 electrons to form harmless water. But now the issue is, suppose oxygen does not find 4 electrons. Suppose oxygen finds 1, 2, or 3 electrons. What does it then form? And what it forms is the oxygen radicals, sometimes called the reactive oxygen species, like superoxide, hydrogen peroxide, singlet oxygen, hydroxy radical. And why are those free radicals so deadly for human beings? Because they don't care where the remaining electrons are located. They're looking for the remaining electrons to form harmless water. If those electrons are in DNA, the DNA will be mutated. If the electrons are membranes, the membranes will fall apart. So those free radicals that are being formed, because they only find a few electrons, they're looking for the remaining electrons to form harmless water, and they don't care where those electrons are located. All right, now the important feature is, in the last few seconds, you just took in a breath of air, and in that air, 18% of it is oxygen. So you can imagine right now, oxygen is now coming into your system here, and most of the rest of that is nitrogen, that breath that you just took in. So about 18% of that is oxygen. That's atmospheric oxygen. Now let me ask you this. So when you breathe in that oxygen, and you can imagine, as every few seconds you're breathing in liters and liters of oxygen, and when that oxygen comes into your system, and it has the potential of forming free radicals, and then the free radicals then will cause damage because they want to form water. Now the idea here is, when you took in that breath and that oxygen now is being delivered to cells and things, where is most of the oxygen in your body right now? In other words, where is the most potential to form free radicals? You breathed in that oxygen. Where is most of it right now? Let me see your answers. Where is it? Where is it most after it enters the lungs? So when it passes through the lungs, and where is it in what cell types and so on? What cell types is where most of the oxygen is located right now? That's right. It's in red blood cells. So in red blood cells, that's where the most potential to form free radicals. Now here comes a very important concept here. Suppose this represents the red blood cell, and in the interior is hemoglobin. Now, you know from pulmonary physiology is that when these red blood cells get pumped to the lungs, the oxygen tension of the atmosphere is very high, and then the oxygen tension of the atmosphere will then is higher than in the red blood cell, and the oxygen tension now will force the oxygen in the lung area through the bilayer onto the hemoglobin. Now remember, there's no transporter for oxygen. It's just simply the oxygen tension is very high in the lung area, and it forces the oxygen through the bilayer onto the hemoglobin. Remember, oxygen tension is a concentration difference. There's a lot more oxygen on the outside than in the inside. So the oxygen tension now forces the oxygen through the bilayer onto the hemoglobin. Okay, so now this picture here. So that's where most of the oxygen is right now in the body. It's in the red blood cell as the red blood cells circulate. Got a lot more to say about that later on tonight. But the important feature here is when oxygen goes through that bilayer onto the way of the hemoglobin, most students never think about this. But when oxygen goes through that bilayer, what's in the bilayer? And you all say, well, I know what's in the bilayer. It's all those phospholipids. Now remember, what's in the phospholipid? And a phospholipid has two fatty acids. What do you remember about the fatty acids of the phospholipids of the bilayer? Well, you say, well, usually the fatty acids, one of them is a saturated fatty acid and the other is an unsaturated fatty acid, polyunsaturated fatty acid, like arachidonic acid, like linoleic acid, those type of things. Now, what do you remember about unsaturated fatty acids? You say, well, they have carbon-carbon double bonds. What do you remember about carbon-carbon double bonds? Now, I'm going to tell you a term right now that a lot of you forgot from high school chemistry. But what is a carbon-carbon double bond that most medical students forget? What is a carbon-carbon double bond? And remember from organic chemistry, what's a carbon-carbon double bond? It's two carbons that are sharing, what's known as a pi cloud of electrons with each other. A carbon-carbon double bonds are two carbons sharing electrons with each other. That's what a carbon-carbon double bond is. You probably thought you'd never hear about that in medical school. But this is reality. So just picture now, in the membrane of all your cells, including red blood cells, all these polyunsaturated fatty acids with those carbon-carbon double bonds. And those carbon-carbon double bonds are sharing electrons with each other. Now, when oxygen goes through that bilayer, just picture what's happening. Just picture what's happening. When oxygen goes through that bilayer, oxygen is going through an ocean of electrons on the way to the hemoglobin. As oxygen goes through that ocean of electrons, sometimes just like a kid in a candy store, the oxygen sometimes just can't help itself. As it goes to the bilayer, sometimes it sees an electron being shared by two carbons, and then the oxygen then takes the electron, because oxygen is an oxidant, oxidizing agent, electron acceptor. Oxygen takes the electron from the carbon-carbon double bond, and then as a result it forms superoxide, and then the carbon-carbon double bond then breaks, forming other free radicals. And you can imagine, if that's all there was, that begins the cascade of destruction. And if that's all there was, you'd be dead in no time. Red blood cells, there it is. The red blood cells would fall apart in hemolysis, and you'd be dead in no time. The body says, you know, I want to live. And if you want to live, you got to do something about this. And one of the ways the body deals with this is vitamin E. Now, because vitamin E is a lipid, vitamin E would be located in the membranes of all the cells, not just red blood cells. It's in all the cells in the human body. Now, why is vitamin E good for me and you? And the reason why it's good for me and you, because when oxygen, just by chance, just by a random chance, if it takes an electron from carbon-carbon double bonds, forming superoxide, which is looking for 3 more electrons to form harmless water, vitamin E will give up its own electrons to the free radicals to detoxify them to form water. So vitamin E, why it's good for me and you? Because it gives up its own electrons to the free radicals to detoxify, preventing the destruction of the red blood cell membrane here. And so another way to view this is, you can view, why is vitamin E good for me and you? I mean, because it gives up its electrons, it's destroyed. It must be replaced every day. Now, another way to view this is vitamin E is acting as a sacrificial lamb in your membranes. When the free radicals begin, vitamin E says, take my electrons to protect its brother and sister lipids in the membranes. Vitamin E gives up its own electrons and is sacrificed and has to be replaced every day. That's why vitamin E is good. And I want to stress, this is not just in the red blood cell. Any cell that's highly aerobic, neurons, must have vitamin E in the membranes, because neurons are highly aerobic, lots of oxygen in neurons, lots of potential to form free radicals, causing destruction here. So vitamin E is in all cells, including red blood cells, protecting against those free radicals that are being generated all the time, every day, in massive quantities. Now, just to illustrate how many free radicals are being generated, you and I have other antioxidants to detoxify. And these other antioxidants, another example, is catalase. Now, catalase will be located in all cells. And catalase, what it does, it destroys hydrogen peroxide. And so catalase is in cells when hydrogen peroxide is around, looking for a couple more electrons to form water. Catalase will destroy it before it destroys the cell. Another antioxidant is vitamin C. Now, vitamin C, in contrast to vitamin E, vitamin C is water soluble. So vitamin C would be inside cells. And again, vitamin C is acting as a sacrificial lamb, just like vitamin E. Vitamin C is inside cells in the cytoplasm. When free radicals production forms, vitamin C says, take my electrons, protect everybody else, and vitamin C is destroyed and has to be replaced every day. Another potent antioxidant is beta-carotene. Now, beta-carotene is that lipid that you find in squash and yams and carrots. It's the yellow-orange pigment. It's a lipid and it behaves just like vitamin E. It sits in membranes, non-toxic, and what it does, it's a free radical scavenger. When the free radicals begin, beta-carotene gives up its electrons. Now, all of these antioxidants, and there are other ones as well, are in all cells. In all cells. But in the red blood cell, because that's where the most potential to form free radicals and destruction. In the red blood cell only, there is yet another potent antioxidant that you must know we'll talk about tomorrow, and that is glutathione. Now, glutathione is in the red blood cell membrane, in the red blood cell cytoplasm, and it's involved in detoxification of the free radicals in the red blood cell, because that's where most of the potential to form free radicals, because that's where most of the oxygen is, in the red blood cell. Now, as you know, glutathione has a lot to do with G6PD. We'll be talking about tomorrow in these type of things. but you must know glutathione. It's a potent antioxidant here. Now, what I want to get at here is, before I conclude here, is just to illustrate how much free radicals are being generated in the human body every day. You think, what's the big deal? It's tremendous quantities. You would think somebody with vitamin E deficiency, you would think catalase, vitamin C, beta carotene, glutathione, all these other antioxidants would take up the slack. It's not enough. You need vitamin E as well. You would think with somebody with G6PD deficiency, you would think, well, vitamin E, catalase, vitamin C, beta carotene, other antioxidants would take up the slack if glutathione is not being made properly. It's not enough. You're going to have hemolytic anemia in G6PD deficiency because you cannot make enough glutathione. You need all of these antioxidants to detoxify the massive quantities of free radicals being generated in the human body every day. And again, I cannot emphasize enough how much free radicals are being generated, how much destruction they do to the human body every day. And you must be able to detoxify. As an example, another example, it used to be thought that when somebody had a heart attack, an MI, it used to be thought, and you get the necrosis of the heart, it used to be thought that necrosis was due to the lack of oxygen because of an occlusion, that there was lack of oxygen and then the cardiac cells died. It's what's causing the destruction is not due to the lack of oxygen. What's causing the necrosis of the heart after an MI, it's that when the occlusion goes away and the red blood cells bring the oxygen back, the oxygen then forms these free radicals. They get out of hand and they cause destruction of the cardiac cells because all those potential to form free radicals, they cause so much damage to the body, as we'll talk about, because you're breathing in oxygen every few seconds. They're going to cause problems like homocystinemia coming up on Friday, many problems here. So please understand the concept of free radicals. Every few seconds, just picture right now, every few seconds you're breathing in the oxygen. It's required for life. The biochemistry of that is coming up a little bit later. But even though it's required for life, it's also deadly for human life. Here. Okay, now what's coming up next is, so we're going to do two vignettes. We're done with the vitamins here, and so we're going to do two vignettes. If Brad can bring up the selection pod and timer, I'm going to give you 1 minute per question, and hopefully to get both of them correct here. Okay, and here is the first question here. Ten seconds. Okay, and we'll end the poll and broadcast the results. Okay, everybody can see that looks like option D was the about half the students put option D, and option C also was chosen quite often. All right, so anyway, the answer to this question is option D. increased adenylate cyclase activity. Now let me ask you this. What is the what's the pathology that this person has? This should be pretty easy. I think every student should be typing in the same answer. What's the pathology that this toddler has? That's right. She has pertussis here. Pertussis because why? Major clue is that whooping cough. One little phrase, whooping cough. It is where you say that's got to be pertussis. Now on board exams, that's just one jump. They might ask what's the pathology, or they might ask what organism is causing this infectious disease and so on. But they're not asking that. They're saying based on your ability to figure out that this toddler has pertussis, they say the toxin produced by the pathogen responsible for the symptoms has which of the following effects on host cells? And the answer is increased adenylate cyclase activity. Now, why? Let me see your answer here. Why did the adenocyclase activity increase? We talked about it earlier. Everybody should be able to get this because pertussis toxin inhibits alpha I of GI by ADP ribosylation, and therefore alpha I will not work. And what's alpha I supposed to do? Alpha I is supposed to inhibit adenocyclase from making cyclic AMP. And now if alpha I is being inhibited, then adenocyclase is now free. It's free to work. And it's going to start, its activity is increased. It's going to start making cyclic AMP. Now let's go through the other options here. What did we talk about either in physiology or biochemistry or pharmacology? What did we talk about earlier that does option A? That does option A. What did we talk about that does option A? Decreased phosphodiesterase activity in liver. That's right. That's right. Well done, everybody. Well done, Starbucks. That's right. Methylxanthines, caffeine or theophylline in tea. What did we talk about that does option B? Option B. Decreased phosphodiesterase activity in smooth muscle. That's right. That was the sildenafil. By the way, notice here, location, location, location. Notice the first three words are identical. Decreased phosphodiesterase activity. Well, the fourth word also is identical. Decreased phosphodiesterase activity in location, location, location. One says the liver and the other says smooth muscle. And the pharmacological drugs that affect them are much different depending on location. Always pay attention to location. Always. I cannot emphasize enough. You must know where things are happening here. Now, option C. What did we talk about earlier that decreases translation by inhibiting elongation factor 2? That's right, two different toxins. They were either diphtheria toxin or Pseudomonas toxin. Well done. And lastly here, option E. What did we talk about earlier that does option E? Increased calcium release. I'm talking about the direct. What directly increased calcium release in smooth muscle? Directly. Let me see your answers here. Not diacylglycerol. There we go. Some of you got it right and some of you did not. And the answer is it's IP3. IP3 opens up the channel to allow calcium release in smooth muscle. Let me ask you this without looking in your book. Don't go looking in your book yet. But the idea is, what's the name of the receptor that was triggered ultimately to do option E in smooth muscle? What was the name of the receptor? Specific receptor. The receptor, that's PIP2, is not a receptor. Not a tyrosine kinase. No. What's the name of the, that's insulin issues, Mithra. And there it is. Now we're getting somewhere. Hilal and Hector, that's right. And the receptor is alpha 1 adrenergic receptor. So alpha 1 adrenergic receptor in smooth muscle. So when epinephrine binds the alpha 1, in turn, it then turns on what? Without looking in your book. Let's go through the sequence of events without looking in your book. When epinephrine binds to alpha 1, in turn, what was activated? That's right. It was GQ, trimeric G protein called GQ. Alpha Q in turn binds GTP and in turn activates what? What does alpha Q turn on without looking in your book? What? Not PI, not IP3. No, that's right. It turns on an enzyme called phospholipase C, and phospholipase C in turn cleaves what molecule? What molecule does phospholipase C cleave? What molecule? There we go, PIP2. And it cleaves it right down the middle, generating two fragments, diacylglycerol and IP3. And IP3, once it's formed, does option E. It releases calcium from the ER. And then the diacylglycerol, along with calcium, turns on what? Diacylglycerol and calcium turn on what again? Diacylglycerol and calcium turns on protein kinase C. There it is. There it is. Now again, I went through, we talked about that yesterday. This should be mastered by now. You should be able to verbalize a sequence of events without looking in your book. Don't go looking in your book. Once you think you mastered it, go through it over and over, 2 or 3 times, then, oh yeah, now I got it, I don't have to hesitate. I can tell you it's alpha 1, which then turns on GQ, which turns on phospholipase C, which cleaves PIP2, down to diacylglycerol and IP3, and so on. You should be able to verbalize that. If you can't verbalize that information, or if you're stumbling, or if you don't, then you have to go back. Don't make excuses. You can do it. You can do it. You just have to do it. You know, you just give yourself credit. You can do it. All right. So again, I went through that exercise to show you, in case you were following behind, you must, that's high yield endocrinology here. You must know the information. All right, now the next question, and the last question here, based on stuff we talked about. Here it is. If Brad can get ready with the selection pod and a timer for 1 minute, here is the next question. Thank you, Brad. Ten seconds. Okay, and there's the minute, and end the poll and broadcast the results. There we go, end the poll, and looks just as predicted. Every time I do this question, it's usually between option C and D, and usually that's what most students put, either option C or option D. And the answer to this question is option C, the 1-alpha hydroxylase here. All right, now let me ask you this here. What is the, now again, most people get the pathology name. What's the name of the pathology? What's the name of the pathology in this 14-month girl? What's the name of the pathology? What's her problem? Everybody should be able to get that. That's right. It's rickets, right? That should be pretty easy. That's the first jump, is you say it's rickets. Now, how do you know it's rickets? Now, remember earlier I mentioned to you, always pay attention to the first phrase. And it says, in March, a 14-month-old girl had a seizure and brought to the ER of a local hospital in Chicago. What do you think the clue is there? March in Chicago. What is the clue with this girl? That's right. You can imagine. I don't know if you ever, I know some of you live in Chicago right now, but some of you have never been to Chicago. In January and February, it's too cold. You don't go outside in Chicago in January and February. So you can imagine by March, this 14-month-old girl has not been outside, and she has not been exposed to sunlight by March. For several months, she has not been exposed to sunlight. Therefore, she is prone to vitamin D deficiency here. And it's confirmed by the radiogram here, the bowed legs and so on, all the characteristics of rickets here. All right, but they're not asking you what's the name of the pathology. What they're going at here is they're saying the active form of the vitamin D now, is most likely deficient in the child, is produced by which of the following enzymes? And the answer is option C. Now let me ask you this. Option C, the 1-alpha hydroxylase. Where is that enzyme located and what turns it on? That's the main enzyme in vitamin D synthesis, is option C. Where is it located and what turns it on? That's right, it's a renal enzyme and it's turned on by PTH to make calcitriol here. Okay, so now let's go through the other options. Option A. What does calcitonin do again? What does calcitonin do specifically? We talked about it yesterday without looking in the book. Don't go looking in the book. But what does option A do? That's right. That's right. It prevents the osteoclast from demineralization of calcium from the bone. Option B. Where is option B located, and what does option B need to work? Where is it located in the tissue, and what does it need to work? That's right. Well done. Well done. Give everybody a chance. That's right. It is a hepatic enzyme, liver enzyme, and it needs vitamin K to carboxylate several blood clotting factors. All right, well done. Option D. Now, that's the one where a lot of students put option D. Where is that enzyme located, and what is its function? What does it do? Yeah, it puts a hydroxygroup of carbon 25, but what's the function? It's a liver enzyme for the purposes, that's right, IFOSA, that's right, for the purposes of making the storage form of vitamin D. Here, well done. So the storage form is made in liver and eventually shipped off to the adipocyte for storage. And lastly, option E, lysooxidase. What does that enzyme need to work, and what does it do? Not vitamin C. Vitamin C is the lysohydroxylase. This is lysooxidase. So again, yeah, there it is. There it is. It is copper. So it requires copper to oxidize hydroxylysine to enable the collagen to cross-link there. Okay, well done. And if you said vitamin C, it's okay. It's okay. I'm glad you say it. This is a time to make mistakes. If you make a mistake, it's okay to do it now. Yeah, no problem. You're bunk friends. Okay, you make a mistake, you'll never forget it. You don't want to make the mistake on the boards, right? This is the time to make mistakes. That's why I went through this series of questions just to see if you can recall things. If you got the wrong answer, it's okay now, not later. Okay, everybody, so we're done with these two questions here. So now what we're done, what we're going next is chapter 11. Now, chapter 11 is it is an overview chapter, a big overview of metabolism. Now, let me say something here. Is that we're just about there. Where's there? Metabolism is where there is. All I did in the first 7 sessions that I've been here is setting the stage. All that molecular biology, all that stuff on genetics with chromosomes and this and that, all the stuff earlier on Saturday as well as yesterday on enzymes and hormones and vitamins. All we're doing is setting the stage for metabolism. Now, the individual metabolic pathways will begin in chapter 12. But chapter 11 is overviews of metabolism, big overviews. And because what I'm getting at is you will never understand individual pathways unless you understand the big picture of metabolism. So that's what chapter 11 is about, is what is metabolism all about in terms of big pictures. What's going on here? in overview format here that you must understand. Understand the bigger picture first. What's going on in the body here? Okay, now now what I mean by this is what happens is a lot of medical students, what they want to do is they want to learn the individual pathways first, and then they want to work their way up to the bigger picture. That's the wrong thing. That's the wrong way to do this. The best way to do this is to understand metabolism, to get a handle on metabolism, is first ask, what condition is the body in? Are you eating a high carb meal right now? Are you fasting right now? Are you eating a high fat meal? Are you exercising right now? Do you have some particular pathology right now? What condition is the body in? And once you determine what condition the body is in, the body always responds to that condition by secreting one or more hormones. There is never a time in the human body where you're not secreting at least one hormone. One or more hormones are always being released depending on what the condition the body is in. And then that hormone then targets a particular organ or organs. And then in that target organ or organs, then you ask what pathways make sense of being turned on and off. And in those pathways, what enzyme is being turned on or off by that particular hormone. So start at the big picture and work your way down to the individual metabolic pathways. And if you do it that way, I'm telling you right now, metabolism becomes alive. It starts to make sense. Always ask yourself, what condition is the body in? Once you determine that, you say what hormone in endocrinology is being secreted in response. Then you say, what's the target organ of that particular hormone? And then in that organ, what pathways are being turned on and off logically? And then what enzymes in those pathways are being turned on and off? And if you do it that way, I'm telling you right now, things become alive. Oh yeah, now I know what's going on. Now I know. And that's what this chapter 11 is about. Understanding big pictures. Approach it with a big picture. And if you do it that way, you'll want to know this information. And things will make sense. All right, now what's coming up here is an overview. Is this figure here on page 160. Now, before we talk about this particular figure, what I want to do first is talk about the all-important glucose tolerance curve. That's not in your book. But you must understand the significance of the glucose tolerance curve. In other words, in this overview of catabolism, the glucose tolerance curve is, I'm telling you right now, it's so critical to the human body. In other words, if you were to eat a donut right now, or a piece of chocolate, or something like that, and as a function of time, measure the blood glucose as a function of time, what would the blood glucose look like? Now, as we talked about earlier, that and as a physician, you got to know the glucose tolerance curve. And as we'll talk about later on in metabolism, the body centers its hormones on the glucose tolerance curve. You must understand the glucose tolerance curve, because in a few minutes, I'm going to ask you, what does the glucose tolerance curve look like for a diabetic? Since we're getting in the area of carbohydrates, anytime we can talk about diabetes, we will. Because diabetes, in my opinion, is the single most important disease for the national boards. You are undoubtedly going to see at least one or more questions about diabetes on the boards. You must understand diabetes. But before we get to that, you'll never understand diabetes unless you understand the glucose tolerance curve. All right, so if you're a normal individual and you start eating at time zero here, if I start eating a donut, what would the blood glucose look like? And you would say, well, the blood glucose will start to rise, reach a maximum, and come back down to normal. And a person would be transiently hyperglycemic. glycemic here, and that's all normal. So the blood glucose would rise, reach a maximum, and then the blood glucose will slowly start coming back down to normal here, and that's all normal. But now the all-important x-axis here. When does it reach a maximum and when does it come back down to normal? You got to know this as a physician. Let me ask you this. Here it comes. When does the blood glucose reach a maximum? If I ate a donut right now, when would my blood glucose reach the maximum? Minutes, hours, days, or give me a time here. When does it reach a maximum? And it reaches a maximum in 45 minutes. So 45 minutes, it reaches a maximum. And then an hour and a half after the last bite of a high carbohydrate meal, after the last bite of a high carb meal, after the last bite of a donut or whatever it is, 90 minutes later, the blood glucose should be back down to normal here. So again, this magenta arrow here, see this magenta arrow? That when the blood glucose comes back down to normal, at 90 minutes after the last bite, it's back down to normal here. Okay, now how high does it go at 45 minutes? It depends how much carbohydrate that you're eating. If you eat one donut, your blood glucose might look like this. You eat 2 donuts, they'd be higher than that. 3 donuts be higher than that. 4 donuts higher than that. But so it reaches a maximum in 45 minutes, no matter how many donuts you eat after the last bite. And then it comes back down to normal 45 minutes later, for a total of 90 minutes after the last bite of the high carb meal. It's better be back down to normal. And how high does it go? Sometimes called the glycemic index. Depends how much carbohydrate that you eat. Your body will absorb all of the carbohydrate that you eat, all of it. Why? It doesn't know when you're going to eat next. Your body doesn't know when your next meal will be. So therefore, you will absorb all of the carbohydrate that you eat. And it reaches a maximum in 45 minutes and comes back down to normal at 90 minutes after the last bite. So this is by the way, now notice, when the blood glucose comes back down to normal, it doesn't keep going down. It's being maintained. And this magenta arrow at 90 minutes is so critical to the human body. I cannot emphasize that enough. So critical to the human body. What I'm driving at is the metabolic pathways going on in the body to the left of the arrow are much different than to the right of the arrow. When the blood glucose comes back down to normal, it doesn't keep going down. It's being maintained, and the body is turning on other pathways to maintain blood glucose here. Critical item that you must understand. Now, speaking of understanding, here comes a question for you. If you were asked, what's responsible for the blood glucose coming back down to normal? Let me see your answers. What's responsible for the blood glucose coming back down to normal? Okay, now everybody's going to say insulin. Now let me ask you this. Is insulin solely responsible for this blood glucose coming back down to normal? You must understand it because diabetes is coming up next when you don't have insulin around. So is insulin solely responsible for this blood glucose coming back down to normal? And the answer is no, it's not solely responsible for that. Why? Because you have insulin independent cells in the body, always removing glucose out of the blood. Like what? Like the brain cells, like red blood cells, and so on, that are always removing glucose out of the blood independent of insulin. So insulin has a lot to do with this, but not solely to do with this here. So it's a lot to do with it, but it's not solely responsible for the glucose coming back down to normal. All right, now this is the normal glucose tolerance curve. Now the issue is, what does the glucose tolerance curve look like in a diabetic? Now let me say something here before we answer this. Now type 1 versus type 2 diabetes. Now type 1 diabetes is juvenile diabetes. That's where there's something wrong with the pancreas not secreting insulin properly. So not enough insulin is being secreted, type 1. Type 2 diabetes, the more common one, that's usually due to something wrong with the insulin receptor. Either not enough receptor, and if somebody's obese, there's not enough insulin receptor around. There may be insulin around, but not enough receptor, and therefore you're not going to get the insulin response. So there's usually, in type 2 diabetes, there's usually something wrong with the insulin receptor. Not enough of it, not enough binding of insulin to the receptor, insulin resistance, those type of things. But when you think type 2 diabetes, you think something wrong with the insulin receptor. So in this review over the next several days, when I use the term diabetes, I'm referring either type 1 or type 2 diabetes, I'm referring to not having the insulin effect, the downstream insulin effect. Either due to not enough insulin around or not enough receptor around, you're not getting the insulin effect. What is insulin supposed to do biochemically? All right, so here it is. In diabetes, type 1 or type 2, if a diabetic eats a donut, then as a result, because they're diabetic to begin with, they're hyperglycemic, the blood glucose will be elevated. And so then to begin with, and then if they start eating that donut, the blood glucose starts to rise high, and then at 45 minutes, then what happens to the rest of the curve in a diabetic situation? Does it stay elevated, or what happens to it? Does it stay elevated? No, it starts slowly coming back down to normal, but at a much slower rate. Why? Because of insulin-independent cells in the body always removing the glucose out of the blood. Slowly but surely, the brain cells, the red blood cells, the pituitary gland, and so on, always removing glucose out of the blood at zero order kinetics. Remember the transporter, GLUT1 or 3 we talked about the other day? Removing glucose out of the blood at a very steady pace, independent of insulin. And so it slowly comes back down trying to come back down at a much slower pace. But before it ever reaches normal, a diabetic gets hungry. After several hours, a diabetic's going to get hungry and the blood glucose shoots back up. And then slowly starts coming back down, and before it ever gets back down to normal, diabetic gets hungry again, and it shoots back up again. So that blood glucose in the blood stays elevated for extended periods of time. As we'll talk about after the break, that extra glucose in the blood is going to be the basis of all of the pathology of diabetes that's going on in the vasculature. All this extra glucose in the blood. See that difference between the normal curve and the diabetic curve? For hours and hours and days. That extra glucose in the blood is going to cause havoc to the vasculature, as we will talk about after the break. Okay, now what we're going to do is we're going to take a 10-minute break right now. It's 7 minutes to the hour, and then come back and then do these. There's 3 overview figures going to come up, and then we'll start to get into individual metabolic pathways. So let's take a 10-minute break, and I will see you in 10. Okay, welcome back for hour 2. Now, where we're at is on page 160 and this overview here. Now, what this overview is about, one time I asked, out of clear blue sky, I asked my wife, I said, why do you eat? And she said, because. And I said, no, seriously now, why do you eat? And she said, let me think, because. And I said, no, seriously now, seriously, why do you eat? And she said, because. I said, I said, come on, man. I mean, I said, you can go watch your Judge Judy, and read a book for a change. The idea is, why do you eat? Some of you at the break, you went out and got something. So the idea is, why do you eat? I mean, a simple question like that, from a simple guy like me. Why do you eat? And why do you eat is right there on page 160, right there in front of you, right there is why you eat breakfast, lunch, and dinner. Because the idea is that when you eat a high carb meal, high protein meal, or a high fat meal, what happens is that food enters your mouth and then eventually gets into your intestine, and then you absorb the carbohydrates, proteins, and fat. And then what happens to the carbohydrate? The carbohydrate the glucose enters all the cells in the body, and the glucose goes to a pathway called glycolysis, coming up a little bit later. And glycolysis, there is no carbon loss in glycolysis, and you generate 2 molecules of pyruvate. Each one is 3 carbons, no carbon loss. But then pyruvate sees a major enzyme in this overview that removes 1 of the 3 carbons of pyruvate as CO2, then generating acetyl-CoA as a product. Now, if you eat a high protein meal, obviously you're not going to McDonald's for that. But if you eat a high protein meal by eating a piece of chicken, something like that, then you break down that protein to amino acids. The amino acids go through metabolism, generating the primary product also is acetyl-CoA. And then if you eat a high fat meal, by the way, fat means triglycerides, not cholesterol. Cholesterol is not fat. Cholesterol is a lipid, but not fat. So if you eat a high-fat meal, all those triglycerides, what happens is then you break down the triglycerides, the 3 fatty acids. By the way, later on this week, we're going to talk about the Atkins diet. I'm not sure if you know what the Atkins diet is. It's that high-fat, no-carb diet. Is it a good diet or not? We'll be talking about that several days from now. But the idea is, when you eat a high-fat meal, you break down the triglycerides at 3 fatty acids. The fatty acids go through a major pathway of life called beta oxidation, in which electrons are removed from the beta carbon, and you generate multiple acetyl-CoA. So notice that acetyl-CoA is a focal point of metabolism in this overview, coming from carbohydrates, proteins, and triglycerides. And then the acetyl-CoA, the 2 carbons, go through in a major pathway for life called the TCA cycle, also called the Krebs cycle, the tricarboxylic acid cycle, citric acid cycle, whatever you want to call it, different strokes for different folks. And in this cycle is where you have enzymes that systematically and in an orderly fashion pluck out the high energy electrons of the acetate group, and you blow off both carbons as a dead end CO2. And what happens to the high energy electrons that are plucked out of the carbon? Well, they're put onto mobile carriers called NAD to form NADH, and FAD to form FADH2, and then they will go through the electron transport chain, and in the electron transport chain, this is where the energy is separated from the electron. And by the time those electrons go on to the oxygen that you breathe in every few seconds, and you must deliver by the erythrocytes. A lot to say about that tonight. But you must deliver the oxygen, because when those electrons go on to the oxygen, they are low energy electrons. Initially, they were high energy electrons, especially when in the carbon of the food that you ate, high energy electrons. But in electron transport chain, that's where the energy is separated from the electron. And when they go on to the oxygen that you breathe in, they are low energy electrons.
Then you ask, what happens to the energy when it was separated from the electron? Well, it's used, half of it is then used to phosphorylate ADP to form ATP. So about half of the energy is captured of that electron coming from the carbon of the food that you eat. That is amazing. Half of the energy is captured to make ATP. And then the other half of the energy is given off as heat. Now, let me ask you this. Is that wasted heat? Is that wasted heat? So half the energy is captured and half of it's given off as heat. And the answer is no, it's not wasted. That's being used to maintain your body temperature at 98.6 Fahrenheit or 37 degrees Celsius. And so the idea is, now that's very efficient. I don't know if you know this, but those of you who were engineers before you went to medical school, the human body is much more efficient than any machine that a human being can make. 50% of the energy of the electron, of the carbon of the food that you ate is captured to make ATP. The other 50% is given off as heat. The best human-made engine, for example, your car, you capture only about 18% of the energy of the octane that you put in your car. 18% is captured to turn the wheels of your car, and the 82% of the energy of the octane is given off as heat. That's why after you run your car, you cannot open up your front hood and then use your hand and touch your engine. You'd burn your hand. You burn your hand if you touch the engine because it's too hot. All the energy is given off as heat. But the human body, 50% is captured, not 18%, 50%. It is incredibly efficient, is the human body. This is why you eat, because you want the energy. You want the energy of the electron, of the carbon you eat to make ATP. And then what do you need the energy for? Obviously to do muscle contraction. Every step that you take is trillions of ATP. Every beat of the heart is trillions of ATP. And so muscle contraction alone consumes huge amounts of ATP. And where is it coming from? You're looking at it on page 160. Also, biosynthetic reactions, making things in the human body. When you make things in the human body, making proteins, making glycogen, making whatever, collagen and things, it costs energy. Biosynthetic reactions cost energy. What else do you need the energy for? Active transport systems. You don't have to know this number, but those of you who are going to be nephrologists, 70% of the energy generated by the kidney is solely used for active transport systems. So that's a lot of energy just used for active transport. So what I'm driving at is that human body needs so many ATPs, trillions and trillions of ATP per second for muscle contraction, biosynthesis reactions, active transport systems. And where is the ATP coming from? Page right here, page 160, the overview of catabolism. Why you eat. As simple as I can make sense out of it. There it is. You want the energy. And why you got to breathe in the oxygen? To suck up those electrons here. And because the energy will be separated from the electron. All right, now what's coming up next is another overview figure with more detail of what happens when you eat a high carbohydrate meal. Now what we're looking at here is the red blood cell. In the middle is the liver. On the right side is the brain, the muscle at the bottom, and the adipocytes on the left side. So the idea is that when you eat a high carbohydrate meal, if you just ate a donut right now, if you just ate a donut, then the blood glucose is going to rise. And when blood glucose rises, because glucose is polar, it cannot go through a membrane, you need those GLUT transporters to get the glucose into a cell. And in the red blood cell, as well as the brain and most cells in the body, there is the GLUT transporter called GLUT1. There's the related one called GLUT3. And this is the transporter we talked about the other day that has that very low Km value of 1 millimolar. Now, let me ask you this. What was the significance of that low Km value again? You can memorize that. You can memorize all you want, but unless you understand why. What's the significance of the low Km value? Easier for the glucose to get in. Well, it's true. More affinity and easy to pass. There you go. Hello. There it is. It is zero order kinetics is the significance. Zero. It doesn't matter what you eat or don't eat. Glucose is entering those tissues at a very steady pace. The significance is zero order kinetics. Again, meaning glucose is always entering, no matter if you eat a high carb meal or you're not eating. It's getting in at a very steady pace in those tissues. Now, once glucose enters the red blood cell by GLUT1 or 3, it goes through glycolysis, getting a few ATP, and because there's no mitochondria in the red blood cell, pyruvate is dead end. It converts to lactic acid, gets dumped into the blood, and picked up by the liver and further metabolized. In the brain and most tissues in the body, glucose enters by GLUT1 or 3, goes through glycolysis down to pyruvate, getting a little bit of ATP. And because there are mitochondria in the brain, the 3 carbons of pyruvate are completely aerobically oxidized to CO2 and water, generating a lot more ATP. How much ATP? A number you don't have to know for boards, but anywhere from 36 to 38 ATP is being generated in most tissues in the body by complete aerobic oxidation of glucose to CO2 and water. And in the brain, that ATP obviously is being used for active transport systems and all those membrane potentials. All right, now in the liver, there's yet another transporter, and it's not GLUT1 or 3. The transporter there is GLUT2. And remember, GLUT2 has that very high Km value of 15 millimolar. All right, here it comes. You know I'm going to ask you this. Get ready. Major item. What's the significance of that very high Km value? Everybody should be getting this. You can memorize. What's the significance? That's right. First order kinetics. And what was the significance of first order? What's the meaning of that? What's the significance of first order kinetics? And the answer is proportional is the significance. Meaning, no matter, when you eat a high carbohydrate meal, when the blood glucose rises, proportionally more glucose enters the liver. The more glucose in the blood, the more glucose enters the liver by first order kinetics. Because there it is. That's the significance. Whereas in the red blood cell in the brain, glucose is getting in at a very steady pace. But the more glucose in the blood, the more glucose now heads into the liver. And in the liver, all this excess glucose is now pouring into the liver. And what's the liver going to do with that glucose? Well, it's going to go through glycolysis to generate energy, 36 to 38 ATPs. And then in the liver, what's that energy going to be used for, the ATP? Well, it's going to be used primarily in the liver for biosynthetic reactions. And it can be used to synthesize cholesterol, for example, to synthesize fatty acids, for example. But also this glucose, when it enters, can then be stored in glycogen. Talk more about glycogen on the next figure. And so you have about, you can store some glycogen in here in the liver. But in the, but all the excess glucose that you eat is, by the way, let me stress here, the amount of glycogen that you make in the liver as well as in the muscle is limited. You and I only have about 1 pound of glycogen in the human body. If you weigh 150 pounds, the amount of glycogen in the body is about 1 pound. And that's roughly distributed between the liver and the muscle cells. A little bit more in the muscle because it's larger than the liver. But you have about 1 pound of glycogen in the liver and the muscle together. about a half a pound roughly in each organ there. Okay, now, but the amount of glycogen is limited. Now that's you do not make more than 1 pound of glycogen. You know those athletes out there who run a marathon. What they might do is do something called carbohydrate loading. What they might do is eat before they run a marathon. They might eat a bowl of pasta, thinking in the carbohydrate loading, they might make a lot more glycogen. They might make 1.05 pounds of glycogen, but not much more, because the body limits the amount of glycogen. Here comes the why question. Why does the body limit the amount of glycogen? We know it uses storage of glucose, that's true. But why does it limit the amount of glycogen? Why doesn't it make more than 1 pound of glycogen? The size, the place, there you go. Now, Omar, there it is. That's the main reason, Omar, that's right. And the reason is, and those of you said take up space, that's true, storage problems. But the main reason of storage problems is because glycogen, with all the glucoses in it, must be hydrated. Those hydroxyl groups on glucose and glycogen, they must have water accompany them. And therefore, it would take up too much space if you make more than a half a pound in the liver and a half a pound in the muscle. It would take up too much space, and it's because it has to be hydrated. A lot of water accompanies glycogen. And you must know that. If you understand that simple biochemical fact, that glycogen must be hydrated. If you understand that, then we'll get in, once we get into the pathologies of glycogen storage diseases later, in the glycogen storage diseases, when you can't break down glycogen properly, and glycogen accumulates, and then you're going to see these words called hepatomegaly, cardiomegaly, because the issue is there's too much glycogen or glycogen stuff in the liver and the cardiac cells, and it takes up too much space, and therefore you get the hepatomegaly as well as cardiomegaly in Pompe's disease. It makes sense if you know the biochemistry here. So the body knows to limit the amount of glycogen because it takes up too much space. And so what the body does here is it'll convert the glucose in the high carb meal that you eat down to acetyl-CoA in glycolysis and so on. And then the acetyl-CoA is then used to make fatty acids. And fatty acids then used to make triglycerides. And then triglycerides are then shipped out to the liver in a particle called the VLDL, very low density lipoprotein. We'll be talking about a couple of days from now. All right, and here comes the question, and then the triglycerides are shipped off to the adipocyte. But now here comes the question. The why question. Why is your body making triglycerides? It converts the carbon of glucose to the carbon of fatty acids and then ultimately to triglycerides. Why? Why? Storage problems, energy storage. Yes, it's an energy storage, that's true, but it's not answering why is it making triglycerides and versus glycogen. Glycogen is a storage form, but why is it making triglycerides here? In excess amounts. It's not limited. You can make excess amounts of triglycerides, but why is it making triglycerides? And here comes the answer. Because triglycerides are anhydrous. There is no water accompanies the triglycerides. That's what the body is doing. It's taking the carbon of glucose, and to store it, it can replenish glycogen, but it's limited. And so what the body does, it converts the carbon of glucose into the carbon of triglycerides and ships them off to the adipocyte for long-term storage. And there is no water accompanies the triglyceride because it's anhydrous. So the carbon has a lot of energy in it, but no water accompanies the triglycerides. And it can be stored in unlimited quantities. You know this, in unlimited quantities in the adipocyte. Beautiful. Understanding why it makes a triglyceride in the first place. Why is it making a triglyceride? Because there's no water accompanies it. It's anhydrous. That's why the body makes a triglyceride in the first place. Simple question, simple answer there. which a lot of students never thought about. All right, but anyway, so these triglycerides, again, are packaged in VLDL. Now, let me say something about triglycerides as well. In VLDL, as well as triglycerides coming from a diet in chylomicrons. Now, triglycerides, what you must understand about them is they cannot go through a membrane. They are too greasy. They cannot go through a membrane. Remember, on the membrane, they're on either side of the membrane. It's a polar region. Remember all those head groups like choline and inositol and all those? So it's polar groups on the edges of the membranes. So triglycerides cannot go through them. So what I'm driving at is triglycerides in the bloodstream coming from chylomicrons or VLDL that made them. Chylomicrons coming from the intestine, VLDL coming from the liver. What happens is that triglycerides must be broken down in the blood. They're broken down to 3 fatty acids and glycerol. And the fatty acids can be absorbed. Fatty acids can go through a membrane, and after they are absorbed in the adipocyte, they are then remade into triglycerides and stored in vesicles in the adipocytes. So there it is. So the triglycerides are made, shipped out, and then in the bloodstream they are broken down to fatty acids and glycerol, and the fatty acids are remade, absorbed, remade in triglycerides and stored in vesicles. Now the glycerol, after they're broken down in the bloodstream, the only organ that can deal with blood glycerol is the liver. So the glycerol heads back to the liver and further metabolized. Now let me ask you this, not shown on this figure. What is the name of the enzyme that situates, it's actually located on the endothelial cell that lines the adipocytes, that breaks down the triglycerides to fatty acids and glycerol? What's the name of the enzyme that breaks down the triglycerides in the bloodstream? Lipase, not hormone-sensitive lipase. I need you on the next page. Now that's right. This is lipoprotein lipase, LPL. Lipoprotein lipase breaks down triglycerides in the bloodstream, coming from the from VLDL and chylomicrons. Now it turns out that lipoprotein lipase is hormonally regulated. What hormone makes sense to you turns it on? What hormone makes sense to you? That's right. That's right. Got to be insulin, right? So insulin turns on LPL at the gene level. So you turn it on. Remember the RAS protein? We talked about insulin yesterday. So you turn it on gene expression, and in the adipocyte, it turns on the gene for lipoprotein lipase here. Okay, now what's coming up here is in the adipocyte and in the muscle, there's yet another transporter for glucose. And it's not GLUT1, 2, or 3. I wonder what that number is if it's not GLUT1, 2, or 3. Let's call it GLUT4. That's right. So GLUT4 is, and remember, GLUT4 is pre-made. It's pre-made in the adipocyte and the muscle and sits in vesicles inside the cell. And so it turns, now what you need to know about GLUT4, don't worry about the Km value of GLUT4, not important. What is important, it is hormonally regulated, and it's regulated by insulin. Is that when you secrete insulin in response to a high carb meal. Now remember we talked about this yesterday when we were talking about insulin mediation. Insulin will bind to its receptor, the tyrosine kinase class of receptor, and then downstream will activate a PI3 kinase, and the PI3 kinase, when it's activated, will now open up the vesicle to allow GLUT4 to be put onto the surface of the muscle and the adipocyte here. So, and then now, so in other words, if you are asked that when you eat a high carbohydrate meal, insulin will go to the adipocyte and the muscle and says to the adipocyte and the muscle, let my people go. And sure enough, more GLUT4 is put onto the surface of the adipocyte and the muscle in response to insulin here. All right, now I got a question for you. I got a question for you. Because this, you know how the national board can ask you about this. They might ask you, what is insulin doing regarding the kinetics of glucose uptake in the adipocyte and in the muscle? Is it increasing Km, decreasing Km, increasing Vmax, or decreasing Vmax of glucose transport? Of the four possibilities, what is insulin doing regarding the kinetics of glucose uptake here? Look at that. Well done, everybody. Man, that's good. That's good. Well done. That's good. That's good. Hey, everybody, give me a 5, man. Give me 5. Let's fist bump each other. That's right. It's increasing Vmax. Why? The more glucose transporter on the surface, therefore the more glucose enters per unit time. There it is. More transporter or more enzyme, in this case here more transporter, therefore more glucose enters per unit time. Kinetics. Your body manipulates kinetics all the time. It's not hard. Kinetics is not hard. Your body loves kinetics. And you're starting to love kinetics because it's not hard here. All right, the Km doesn't change. That's right, the Km doesn't change. But the amount of transporter, the number of transporters are increasing, therefore more glucose entering per unit time. Notice how you thought your way through that. You didn't memorize that. It just makes sense. All right. All right, now in the muscle cell, the glucose enters, it goes through glycolysis, and in the Krebs cycle, generating a lot of ATP. And in addition, in the muscle cell, that glucose can then be stored in glycogen here. So you replenish that 1/2 pound of glycogen in the muscle cell. Now in the adipocyte, that glucose enters, and then that glucose will be used to make the glycerol platform to put the 3 fatty acids on in the adipocyte here. All right, now another item here. Now you must know this next item. In the muscle only, not the adipocyte, in the muscle only, there's yet another way you can put more GLUT4 on the surface, and that's by exercise. If you go out and exercise, then you put more GLUT4 on the surface. Now the National Board, I have not heard about the mechanism being on the boards. They do ask about exercise and GLUT4, but they never ask about the mechanism, so I wouldn't be too concerned about that. What happens is when you exercise, you're consuming ATP down to AMP. AMP levels rise, and then AMP turns on a particular kinase that opens up the vesicle to allow more GLUT4 onto the surface. So it's AMP dependent kinase is being turned on in the muscle only, not the adipocyte, in the muscle only. So if you so on board exams, so you talk about exercising conditions, you think to yourself, well, in the muscle, you're putting more GLUT4 on the surface. Does that make sense to you? Does it make sense to you? Do you want more GLUT4 on the surface when you're exercising in the muscle? Yeah, right, because the muscle needs energy, needs ATP to do muscle contraction. So it needs fuel to burn, and now glucose is coming out of the blood. It's breaking down glycogen, yes, to use that glucose, but it's also getting glucose from the blood. And so more GLUT4 transporter, more glucose enters the muscle to burn to make ATP. All right, now to summarize all this, notice here how insulin has dominant effects. And the way insulin chooses to exert its effects is one of 3 ways. One of 3 ways. One way insulin can exert its effects is by increasing dephosphorylation of certain enzymes. Remember yesterday in that all-important insulin mediation page, how insulin works. One of the things that insulin does, it activates a phosphatase. And the phosphatase causes increased dephosphorylation of certain enzymes. Another way insulin can exert its effects is by increasing gene expression here. Gene expression, you just, right here, as an example, lipoprotein lipase is turned on at that gene level by insulin mediation. And now the other way insulin can exert its effects, it depends on what pathway we're talking about. But the other way insulin can exert its effects is by causing certain allosteric activators, certain allosteric inhibitors that normally do not function in the presence of insulin. It all depends. What pathway are we talking about? The way the insulin chooses to turn on or turn off a pathway, either at the dephosphorylation level, either at the gene level, or by allosteric activators or inhibitors. It all depends what pathway we're talking about here. I cannot emphasize that enough. Now is the time to talk about what are the insulin-dependent tissues. And as you can see by the space, there aren't that many tissues. In other words, what in the body right now, what cell types have a receptor for insulin? In fact, there are just 3 tissues that have a receptor for insulin, respond to insulin. So here it is. 2 of the 3 tissues that respond to insulin got to be, right, you would say, got to be the adipocyte, and the other got to be the muscle cell, right? The muscle and adipocyte have a receptor. Now, the third and last tissue in the body that has a receptor for insulin. Now, little helpful hint here, little helpful hint. On the board exams, if they ask you, what tissue does this or does that? And if you don't know the answer, and you want to take a guess, and if you don't know the answer, you want to take a guess. If it's a biochemistry question, and you don't know the answer, take a guess and say the liver, because you're probably going to get it right. If it's a biochemistry question. If it's a cardiology question, you don't know the answer, take a guess and say the heart. If it's a neurology question, you don't know the answer, take a guess and say the brain. But if it's a biochemistry question, take a guess and say the liver, you're probably going to get it right there. Okay, so the 3 organs that have a receptor for insulin. All the other organs and tissues are insulin independent. These 3 here are insulin dependent tissues. Now, one thing I want to stress here as well is that what insulin is telling the adipocyte and the muscle to do is much different than what insulin is telling the liver to do. One more time. What insulin is telling the muscle and the adipocyte to do is much different than what insulin is telling the liver to do. Notice here in the adipocyte and in the muscle, insulin has everything to do with glucose uptake. The issue of the GLUT4. Glucose uptake, and it has something to do with glycogen as well and LPL, but insulin has a lot to do with glucose uptake in the muscle and the adipocyte. In the liver, insulin has nothing to do with glucose uptake. Why? It doesn't have the GLUT4, it has GLUT2. It has a different transporter. Insulin has nothing to do with glucose uptake in the liver. Well, then you got to ask, well, what's insulin telling the liver to do? Here it is. Once glucose enters the liver by the GLUT2 transporter, the first order transporter, once glucose enters the liver, insulin takes command of the liver and tells the liver what to do. Insulin tells the liver, drive glycolysis to make ATP, turn on glycogen synthesis, turn on cholesterol synthesis, turn on fatty acid synthesis. How insulin's telling the liver, start synthesizing things, because we are in the time of plenty. We just ate a high carb meal. We are in a time of plenty, a time of excess. Synthesize things. Synthesize glycogen, cholesterol, triglycerides in abundance. And so insulin's telling the liver, make triglycerides in abundance and ship them out in VLDL. While all that's going on in the liver, insulin comes over to the adipocyte and tells the adipocyte, hey, adipocyte, get ready with your LPL. Because I'm telling the liver to make triglycerides. You're coming your way soon. Get ready with your LPL, because you need to break down those triglycerides to absorb the fatty acids into the adipocyte. and how insulins turn on GLUT4 in adipocyte to get the glycerol platform to put the fatty acids on. Notice how insulin's directing the show. Beautiful how insulin's working here. How insulin's telling the liver what to do, and how insulin's telling the adipocyte what to do regarding all these fatty acids and triglycerides and so on. How insulin's telling the muscle, store that glucose in glycogen here. It is beautiful how insulin directs the show. I keep telling you, these hormones are conductors. They're conducting the metabolic orchestra, as illustrated right here regarding insulin. It is incredibly beautiful. Now, if you understand that simplified version of what insulin is doing here, then you could start to understand what's going to go on in diabetes. You can imagine in diabetes when you don't have the insulin effect. The body doesn't know what to do. It's a mess. The liver doesn't know what to do. The adipocyte doesn't know. What am I supposed to do? I ate a high carb meal. What am I supposed to do with this glucose? The body is a mess when you don't have the insulin effect. Now notice over here. Now here comes a very important concept that you probably never thought about this in pathology, when you had pathology. I hate to say this about diabetes, but it's true. Diabetes is an amazing disease from a biochemical point of view. What I mean by that is, notice here, in diabetes, where do you see pathology? Where do you see clinical manifestations, clinical problems in diabetes? Notice you don't see pathologies in the insulin-dependent tissues. You see the pathologies in the insulin-independent tissues and so on. And it's very ironic. And that's in contrast to most other diseases. In most other diseases, you see pathology in those tissues in which an enzyme or whatever is not working. You see the pathology in those particular tissues. For example, in PKU, when you don't have phenylalanine hydroxylase, then phenylalanine will accumulate, phenylketones will accumulate, and then it causes brain problems, mental retardation, and so on. Later on tonight, we're going to be talking about galactosemias. And in galactosemias, two major organs that deal with galactose is the liver and the brain. And when in galactosemias, when you cannot metabolize galactose properly, you're going to have liver cirrhosis and mental retardation, because those tissues is where the defect is. That's true of most diseases. You have pathology in those tissues where the enzyme is not working like it should. But in diabetes, it's not the insulin-dependent tissues that suffer. It's the insulin-independent tissues. So how do you explain all that? I mean, think about that. That is an amazing fact that you probably never thought about. But now why? Why is it? How does the biochemistry explain that the pathology of diabetes is in the insulin-independent tissues? Well, here it is. Here it is. And the answer, by the way, is not hard to understand. Remember that glucose tolerance curve. Remember I mentioned to you that in a diabetic situation, that the glucose stays in the blood for extended periods of time, for hours and days and weeks. If it's not controlled properly, blood glucose, then the glucose stays in the blood. And you should ask, why is glucose staying in the blood? Why is it not entering cells and being metabolized? That's coming up in the next hour. But the idea is that glucose stays in the blood. And when glucose stays in the blood, it's going to cause problems in the bloodstream, causing all of the pathologies of diabetes. Now here comes one of many things that can happen to that glucose. Now you don't have to know the structure of glucose, but glucose has 6 carbons, 5 of which have a hydroxyl group. But it's carbon 1 that is the business part. It has an aldehyde group. So carbon 1 of glucose has an aldehyde group. Now as I mentioned to you this yesterday, aldehyde groups are highly reactive. And aldehyde groups react with amino groups. Amino groups of what? For example, the amino groups of hemoglobin. Now what this represents here is, remember, hemoglobin has 2 alpha and 2 beta subunits. Each subunit has an N-terminus, amino terminus. That's what it represents here, the amino terminus of each of the 4 subunits of hemoglobin. In addition, hemoglobin on its periphery has lysine residues, and lysines have terminal primary amines. So you can envision now hemoglobin with lots of amino groups. So what happens is, as time goes on, glucose in the blood, hemoglobin in those red blood cells, what can happen is glucose can now react irreversibly, non-enzymatically. The aldehyde group of glucose can react with amino groups of hemoglobin. As a result, it forms the product where glucose is now in covalent linkage irreversibly onto the hemoglobin amino groups. And the product is called glycosylated hemoglobin, also called glucosylated hemoglobin, but most commonly called hemoglobin A1C. Here it is. That is hemoglobin A1C. Now let me ask you this. Do you, if you're a normal individual, do you form hemoglobin A1C? Do you have hemoglobin A1C in your bloodstream right now? And the answer is yes. Why? Because you have 5 millimolar glucose in the blood. Because you have 5 millimolar glucose, you also generate hemoglobin A1C, and so do I. But in a diabetic situation, because the more glucose in the blood, hyperglycemia, therefore the more hemoglobin A1C is being formed, and it's irreversible. And it turns out in laboratory values, if hemoglobin A1C is 7% or greater, that's considered a diabetic situation. 7% hemoglobin A1C or greater is considered diabetic. 6% or less is considered normal, but 7% or greater is considered diabetic. Here. All right, now, as you know in pathology, that hemoglobin A1C is diagnostic for the best indicator of diabetes is hemoglobin A1C. Yes, you can measure sugar in your urine and blah, 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 but the best indicator is hemoglobin A1C because it's irreversible. All right, now another item here. Is there anything, to understand the pathologies that go on in diabetes, is there anything special about hemoglobin amino groups to which glucose can latch onto? Is there anything special about those amino groups that glucose has to latch on to those amino groups of hemoglobin? Absolutely not. There's nothing special about hemoglobin. Glucose latches on to the amino groups of hemoglobin, but don't you think it would also latch on to the amino groups of other proteins in the vasculature? Yes. Like for what? Like where? For example, in the kidney, you have all those transporters that are pumping things in and out of the kidney and so on. And those transporters, part of the transporter faces the bloodstream. And as blood circulates by and glucose is in the blood, glucose can latch on to the amino groups of the transporters. And so now you have glucose latched on to the transporters of the kidney. And as time goes on, and we're talking months, if not years, as time goes on, those transporters will start to fail. And when they start to fail, renal problems that are known to go on in diabetes. Not only that, so glucose can also latch on to the amino groups of collagen in your vasculature. Now it turns out that collagen has a very long half-life, like a month or whatever. But when glucose latches onto the collagen amino groups, collagen half-life is extended. It's not turned over as it should be. It stays around longer than a month, maybe 5 weeks or 6 weeks, something like that. The half-life is extended, but the fibroblasts continue to make more and more collagen. The old collagen is not turned over. New collagen is being made. What do you think happens to the capillaries and so on? As time goes on, the collagen, there's so much collagen around, the capillaries eventually start to constrict and causing clogging and so on, constriction of the capillary beds and so on. So as time goes on, what's going to be the problem? Amputations, poor blood circulations and so on, characteristic of diabetes. Not only that, the glucose, because when it stays in the blood, because it's osmotic, it can draw water away from the brain, and in pathology it's called hyperosmolar edema, because glucose is acting like a sponge. It's sucking water away from the brain and causing hyperosmolar edema. All because glucose is staying in the blood and wreaking havoc to the vasculature, slowly but surely with time. Not overnight, but all the complications of pathology, of diabetes, is centered on glucose staying in the blood. And you must ask, why is glucose staying in the blood? Why is it not going into the cells to be metabolized? The biochemistry of that is coming up in a little bit. But you must understand here why diabetes is so complicated, because it's the vasculature is suffering with all that glucose in the blood. Okay, now so this is what on this page here. Got a lot to say about diabetes in the next hour as well. Now this is what happens when you eat a high carbohydrate meal. But the next item is what happens when you're fasting or starving, or what happens when you eat a high fat meal as well, or high protein meal. Then this situation that goes on. Now the idea here is that when the blood glucose drops, again, the body says, I don't want to die. And so because you don't want to die, you're going to send out hormones. I keep telling you, you always say, what condition is the body in? Well, the condition here is fasting or starving or high fat meal. And one of the hormones is going to be released because the blood glucose is dropping is glucagon. So now glucagon, its main target organ is the liver. And in the liver, what happens is the glucagon will come to the liver. It binds to the beta receptor. We talked about this yesterday. By binding to the beta receptor, it then activates GS. Alpha S then turns on adenocyclase to make cyclic AMP, which then turns on protein kinase A, and protein kinase A, among other things it's going to do, one of them is to phosphorylate a major enzyme in glycogen breakdown, and glycogen will be broken down to glucose to try to maintain blood glucose values. But it turns out the amount of glycogen is limited in the liver. Now let me ask you this: How long does it last? How long does the glycogen in the liver last under sedate conditions? If you're sitting down right now at your computer and you haven't eaten anything, how long does your liver glycogen last to try to maintain blood glucose? No, not 2 to 3 days. That's too long. Not 1 day. That's too long. And the answer is 12 hours. You and I have about a 12-hour supply of liver glycogen to maintain blood glucose. 12 hours, it's all it lasts. And now if you go out and walk, your glycogen will last about 2 or 3 hours. If you go out and exercise, your glycogen will last about an hour or less. So it's going to run out here. And but it doesn't last very long. And because it's going to run out, the body says, under the command of glucagon, glucagon will tell the liver, hey liver, start breaking down glycogen and simultaneously kick in gluconeogenesis, the genesis of glucose. Why? Because the body knows that glycogen will run out. So the glucagon will tell the liver, turn on gluconeogenesis. By the way, you should ask, on the previous page, the muscle had glycogen right here. See, on the previous page, you see glycogen in the muscle. You should ask, well, why isn't the muscle helping to maintain blood glucose? You know why that is? You know why the muscle doesn't help out maintaining blood glucose? Because the muscle is a very selfish, greedy organ. The muscle thinks all by itself. The muscle thinks it's all about me, the muscle, and muscle, me. It's all about me, the muscle says. Whereas the liver is a very kind and wonderful organ. The muscle is a greedy organ. It has glycogen, but it saves it for itself. It doesn't care about the other organs. But the liver is a very kind and wonderful and gentle organ. The liver is saying to the community of organs out there, hey, other organs, hey, pituitary gland, you need some glucose over there? Hey, have some of my glucose, because I'm going to share it with you and you and you, because the liver is a very kind and wonderful organ. Whereas the muscle is a greedy, selfish organ. It only thinks about itself. It's a greedy SOB. It's all about me, the muscle says. But the liver is a kind organ. All right, so the muscle has glycogen, but it doesn't share because it's greedy. All right, so now the glucagon will come to the liver and tell the liver, hey, liver, I know you're kind, I know you're wonderful. We all know the liver is wonderful. Don't abuse the liver with things like alcohol or whatever. Don't abuse it. Don't abuse it with chemicals and things. The liver is wonderful. In any case, glucagon will tell the liver, hey, liver, kick in gluconeogenesis. If it's going to kick in gluconeogenesis, you got to ask, where is the carbon coming from that the liver is going to use to make glucose? Because after all, if somebody's starving, no carbon coming into the mouth. You must drink water. We'll explain water later on. You must drink water when you're starving. But if no carbon's coming into the mouth, yet the liver is making glucose, you got to ask, where is the carbon coming from that the liver uses to make glucose? It comes from three sources. One source is lactate and pyruvate. So lactate will come into the liver, convert to pyruvate, and then the pyruvate is then converted in the pathway of gluconeogenesis to make glucose, which gets dumped in the blood to maintain blood glucose at 5 millimolar. Now, a second source and the dominant source of carbon the liver uses to make glucose are the glucogenic amino acids. Now, remember, most of the protein in the body is in the muscle. So what happens is there's muscle protein breakdown, muscle wasting, and then the glucogenic amino acids will funnel their way down to alanine in the muscle. So the glucogenic amino acids will funnel their way down to alanine. Alanine will be dumped into the blood. And remember yesterday or the day before, Saturday, I asked you, what rings a bell? If on board exams they tell you a patient has elevated alanine in the blood, you think starvation conditions because you're breaking down muscle mass. So alanine in the blood will start to rise. Alanine will come into the liver. The amino group is removed to make urea, and the 3-carbon skeleton is pyruvate to maintain blood glucose values here. Now the third and last source of carbon to make glucose. A lot of people are looking over here in the adipocyte and thinking all those fatty acids. Now, one thing I want to stress is it's not the fatty acids. Fatty acids cannot be used, the carbon cannot be used to make glucose. So human beings can take the carbon of glucose and make the carbon of fatty acids. The reverse is not true. We cannot take the carbon of fatty acids to make the carbon of glucose. So it's not the fatty acid. What happens is the triglycerides and adipocytes will be broken down. Now let me ask you this. What's the name? So what happens is under fasting, starvation conditions, what's the name in the adipocyte of that very, very important enzyme that sits right about here that breaks down the triglycerides, the 3 fatty acids and glycerol? Now I need it. There we go. Not lipoprotein, don't say lipase. There are many lipases. Which one? And Samantha, that's right. And Takir, now I need you. And Pfizer, that's right. It is hormone-sensitive lipase because it's sensitive to all those hormones and so on. By the way, glucagon has no receptor in the adipocyte. You would think it does. It doesn't. There's no receptor for glucagon in the adipocyte. It's sensitive to the other hormones like epinephrine, cortisol, ACTH, norepinephrine, and so on, but not glucagon in the adipocyte. But in any case, so the HSL is turned on by those other hormones under fasting starvation conditions, and triglycerides are broken down to 3 fatty acids and glycerol. And it's the glycerol, Remember, the only organ that can deal with blood glycerol is the liver. So the glycerol will head back to the liver. Those measly 3 carbons are then shown right here as well, used to make glucose there. So those are the 3 sources of carbon used to make glucose here. Then you got to say, well, what happens to all those fatty acids if they're not being used to make glucose? Well, what happens to them? Well, what happens to the fatty acids is the vast bulk of them will be dumped into the bloodstream, picked up by albumin. Many of the fatty acids are brought over to the muscle. They go through aerobic oxidation all the way to CO2 and water, generating well over 100 ATPs. Don't worry about the exact number, but it depends which fatty acid we're talking about, but you get well over 100 ATPs per fatty acid. And in the muscle cell, that ATP is being used for muscle contraction, obviously. Now, many of these fatty acids are brought over to the liver, and in the liver, they go through beta oxidation as well. But unlike the muscle, the acetyl-CoA is not further burned. In the liver only, the acetyl-CoA are then converted to, as they start to rise, they then go into yet another pathway called ketone body formation, and ketone bodies are then dumped into the blood, and ketone body levels in the blood will start to rise. Let me ask you this, how many ketone bodies are there? I'm looking for a number. How many ketone bodies are there? That's right, there are 3 ketone bodies, as you know. Only 2 of the 3 are used as a fuel. The third one we'll talk about later on is a byproduct, but it can be used diagnostically. But here comes the why question. Why does your body make ketone bodies from the acetyl-CoA, which comes from the fatty acids after all? Why do you make ketone bodies in the first place? Why? Let me see your hand. Do you know why you make ketone bodies? And the answer is they're energy for the brain, energy for the muscle. It's true. That's what you're saying is true. What you're saying is true. But so are fatty acids. Fatty acids are an energy form or energy fuel. But here comes the answer. The reason why the body converts fatty acids into ketone bodies in the liver to make those ketone bodies, because ketone bodies are a water-soluble fuel, just like glucose. So unlike fatty acids, which are water insoluble, they cannot cross the blood-brain barrier. Whereas ketone bodies, just like glucose, can be transported into the brain. So they are a water-soluble fuel. And when they come into the brain, as well as other organs like the muscle, the ketone bodies convert the acetyl-CoA back to the acetyl-CoA, and by doing so, they inhibit a major enzyme here that we'll talk about later on in next chapter. And then by inhibiting that enzyme, we're now sparing what little glucose is around for the red blood cell. And so that's why you're inhibiting that enzyme, to spare what little glucose is around, namely for the red blood cell here. So namely for the red blood cell. Okay, now what we're going to do is we're going to take a 10-minute break. We're going to take a 10-minute break. I'm almost done with this figure, but I know it's 2 minutes after the hour, so I'm going to come back to this figure and finish up this figure and make some more very critical points. So let's take a 10-minute break, and I will be back. Okay, everybody, welcome back to this big overview of fasting and starvation here. So just to conclude this particular figure, where we left off is we talked about the idea that if you eat if you're fasting and then you're mobilizing triglycerides in the adipocyte by hormone sensitive lipase, the fatty acids, many of them are brought to the liver. The liver now converts those fatty acids into ketone bodies, which are water soluble. They get dumped into the blood, and many of these fatty acids will go into the brain as well as in the muscle cells, and they then get converted back to acetyl-CoA, and by doing so, they inhibit a major enzyme we'll talk about later on. And by doing that, we now spare what little glucose is in the body, and spares what little glucose is in the body for a cell type that can only use glucose as a fuel, namely the erythrocyte. Now we've almost come full circle here. That's why I wanted to make sure I took the break, because this is so important. I know you were getting a little tired, you needed a break, and I didn't want to blow it by you. Remember earlier I said to you, why does the body care? This was like last week sometime. I asked you, why does the body care how much glucose is in the blood? When the blood glucose is too high or too low, the body sends out hormones to fix the problem. And why does the body care? A lot of you thought it was because of the brain. No, the brain can use ketone bodies. It's the erythrocyte. The erythrocyte needs that glucose, and glucose only as a fuel. So that's why the body goes all out to maintain blood glucose with all those hormones like epinephrine and glucagon and cortisol and so on. It's because of the erythrocyte. It can only use glucose as a fuel. So this is, again, sometimes called the glucose sparing effect. Now, not now, but shortly in this hour, I'm going to ask you, okay, well, to come full circle, because we almost come full circle, then why does the erythrocyte need glucose for? If it needs the glucose and the body's going all out to maintain blood glucose, what does the erythrocyte need glucose for? Because after all, a lot of students think that the red blood cell is just a bag of hemoglobin. It's much more than that. It needs glucose. And what's it needed for? It's coming up soon in this hour. You must understand that what's going on in the human body. Okay, now so the ketone bodies, again, are used as an alternative water-soluble fuel in starvation conditions. Okay, now to summarize this particular figure, glucagon has dominant effects, and how glucagon exerts its effects depends on which pathway we're talking about. One way glucagon can exert its effects is by increasing phosphorylation of certain enzymes. Remember all the phosphorylation business by protein kinase A. So certain enzymes are phosphorylated, and by doing so, some enzymes are on, some enzymes are turned off by phosphorylation. Another way glucagon can exert its effects is by increasing gene expression. What genes are being turned on? Gluconeogenesis. Just put page 78. You see glucagon, remember the Krebs protein, turning on a key gene in the pathway of gluconeogenesis. And then the other way glucagon can exert its effects is by causing certain allosteric activators, inhibitors that normally do not function, now function in the presence of glucagon. And it all depends what pathway we're talking about. Which of the three ways glucagon chooses to turn on or turn off a particular pathway here. And it all depends on the pathway. Major. You got to understand this. All right, now before we leave, so we're done with this figure, big overview figure. Now, let me say something here, and I hate to say this, but it's true. Now, if you think about it, over these last two figures, we talked about high carb meal and all this fasting, starvation conditions, and so on. I hate to say this, but this is true. At any one moment in your life, your body is either on page 162 or on page 163 at any one moment in your life. And here it is. For the first 90 minutes of any carbohydrate in your meal, your body's on page 162. Why? Because of the carbohydrate, the blood glucose rises, the body responds by secreting insulin. So therefore your body's on page 162. And it stays there for 90 minutes. And then remember the glucose tolerance curve? Remember the magenta arrow at 90 minutes. And I said this is a critical arrow. At 90 minutes after the last bite of the high carbohydrate meal, the body shifts to page 163. Why is the body shifting to page 163? Because the blood glucose drops below 5 millimolar, and therefore no insulin, the body secretes glucagon. And then glucagon does all the stuff you see here on page 163. And it stays on page 163 for days and days and weeks until you eat another carbohydrate meal. Even if you eat a high-fat meal, even if you eat a high-protein meal, the body's on page 163 because the blood glucose is dropping below 5 millimolar. It is amazing, that glucose tolerance curve. I mentioned to you, you must understand that glucose tolerance curve. For the first 90 minutes of any carbohydrate in your meal, no matter what else you're eating, if there's carbohydrate there, the blood glucose will rise, you secrete insulin. But after that, 90 minutes later, when the blood glucose comes back down to normal, the body says, we're going to send out the glucagon and other hormones as well to maintain blood glucose. It is amazing. You probably never thought of that, but it's true. It is a true statement here. So the body stays on page 163 for days and days until some carbohydrate comes into your meal. Then you eat it, and it switches to page 162. Back to that page. It is amazing. All right, now we'll talk about the Atkins diet later on, but in the Atkins diet, you're on page 163 because there's no carbohydrate, so therefore you're on 163. There's never a time that you're not on either 162 or page 163. Hate to say it. Amazing. If you know a little bit about chemistry, it is amazing. All right, now from here on, now until we're done, on Friday, now until we're done, it's going to be one metabolic pathway after another. And you must, on these pathways, as long as you're paying attention, I'll tell you what you need to know about the pathways and what you don't need to know. And please try to understand what these pathways are doing for you. If you're just sitting there, just going through the motions and trying to memorize these pathways, forget about it. You must try to assemble the pathways into the bigger picture. Try to understand what these pathways are doing for you, and they start to make sense. Oh yeah, now I understand. And now I understand. Now, speaking of a pathway that should make sense, is a pathway that most students despise. And here it comes. And it's glycolysis. All right, now let me say, I get it. I mean, I feel the vibes coming my way. Now let me say something about glycolysis here. And I know what you're probably thinking out there. You're probably thinking, glycolysis, that's a pathway I heard 100 times already in my career. I'm sick of hearing about this pathway. I hate this pathway and this and that. And let me say something about glycolysis. If you never understood this is one of the two most important pathways for human life, is glycolysis. You got to understand glycolysis. If you never understood glycolysis or appreciated glycolysis, it was never taught to you properly. You must understand glycolysis. It's one of the two most important pathway of them all. And now in the pathway is there's 10 steps and 10 enzymes, meaning 10 genes. Now you might think to yourself, I don't hear about glycolysis much in other courses, and so therefore it's not that important. It's too important. The reason why you'll hear about it much in pathology and so on is because any enzyme defect if there's a complete enzyme defect, you're dead. This is a pathway which is essential for life. And if you have any complete enzyme defect, you're cooked. You cannot survive without this pathway. And that's in contrast to most other pathways. For example, later on in this chapter, we're going to be talking about galactosemia and so on. There you can have a complete enzyme defect, but the person still lives. It may not be good life, but the person still lives. Purine salvage. When you don't have when the gene for HTPRT is genetically defective, it may not be good life with all the gout and the chewing of the fingers, but the person still lives. So those pathways like purine salvage, galactosemias, glycogen storage diseases, that's where you're going to see pathology because the person still lives. But glycolysis, you're unlikely to see anything about it in pathology because the person is dead. The fetus is miscarried if it's a complete enzyme defect. Now there will be one pathology coming up in which is a partial enzyme defect. But all the other enzymes, complete enzyme defect, there's no way you can survive without the pathway. Give the pathway the respect it deserves. Glycolysis. Now it's going to take most of this hour to go through this single pathway. Why? It's so important. You're going to see why it's so important. So please don't be like this person where you're sitting there thinking, oh man, this is going to be terrible. He's raining on my parade. You know. Don't be like this person. Have a positive attitude about glycolysis and make sense out of it. If you make sense out of it, it will start, it'll become much more apparent why this pathway is so important for human life. All right, now this pathway, 10 steps, only 3 of them you need to know for the boards. The other 7 steps, 7 enzymes, forget about it. But please write small. So again, when you see the pathway, don't go panicking. I'll tell you what you need to know. So don't panic. Trust me. Don't panic here. All right, now the way I do the metabolic pathways is I always go through the biochemistry first. What do you need to know about the straightforward biochemistry? And then after that, start backing off and integrate with other disciplines. You're going to see the integration of biochemistry with pathology coming up, with physiology coming up on this page. All right, now in terms of the biochemistry, is that when glucose enters the cells, all 10 trillion cells in the human body, glucose will enter by the GLUT transporters. And then once glucose, and by the way, every cell in life, not just in the human body, but every microbe, every plant cell, every cell has glycolysis. And when glucose enters every cell in the human body, right at the beginning, it sees the first of 3 steps you need to know, and there is an enzyme called hexokinase, and the name of the enzyme tells you what it does. Because it's a kinase, it uses the terminal phosphate of ATP to stick that phosphate onto something. What's the something? The first word says hexose. Glucose is a hexose, and by doing so, it puts phosphate onto the glucose, making glucose 6-phosphate as the product. Now, importantly, so this is a pathway which generates energy, generates ATP. Yet, right at the beginning, you lose ATP. Why are you putting phosphate onto the glucose? You must understand the why, because there's going to be pathology later on. But why are you putting phosphate on the glucose? Why? Why are you putting phosphate on the glucose? That's right, everybody, that's right. And the reason is to trap the glucose in the cell. Once you put the phosphate onto the sugar, those GLUT transporters cannot move glucose phosphate out of the cell back into the blood. Please keep that in mind. That's going to be so important later on. Once the phosphate's on the sugar, it's trapped inside the cell. All right, now another item here. Why is that enzyme highlighted in the red box? And because it's regulated. It's allosterically regulated by glucose 6-phosphate, the product. So if the product is not needed, it accumulates, inhibits the hexokinase in the liver as well as the pancreas. But in the liver, there is yet another enzyme. The liver has hexokinase, but it has another enzyme that does the same reaction, and it's called glucokinase. That because it's a kinase, it uses ATP to phosphorylate glucose, trapping the glucose in the liver. Now it turns out this enzyme, glucokinase, you need to know it, why it is hormonally regulated. Here it comes. What hormone makes sense to you turns it on? What hormone? Yeah, and the answer is, that's right, it's insulin. Insulin turns it on at the gene level. So insulin chooses at the gene level to turn on glucokinase. Now, why is that? Now, think about it. You can memorize, but why does that make sense? Because we just talked about earlier. I'm going to talk about it again and again and again in metabolism. When you eat a high carbohydrate meal, all the excess glucose funnels to the liver by the GLUT2 transporter, first order kinetics. And once glucose enters the liver, then insulin takes command of the liver. And one of the things it's going to do right at the beginning, it's going to tell the liver, turn on glucokinase to trap all the excess glucose in the liver. There it is. So it's trapped now in the liver in response to the high carb meal. All right, now let's proceed here. Okay, now we'll come back to the step. We're going to come back to the step a little bit later in some disease called, what's the name of that disease called again? Oh yeah, diabetes. Now let's proceed. Now once glucose 6-phosphate is formed, it sees a low yield enzyme, forget about it, that makes it into fructose 6-phosphate. Now once fructose 6-phosphate is formed, it sees the top enzyme of glycolysis. And it's always on the board, it seems, and this enzyme is called PFK1, which is abbreviation for phosphofructokinase 1, PFK1. Now, if there's a PFK1, that means there's going to be a PFK2, and there will be before I'm done with the figure. Now, what does PFK1 do? Because it's a kinase, it means it's going to use ATP, because it's a kinase, to phosphorylate what? To put the phosphate onto phosphofructose, the substrate, making the product fructose 1,6-bisphosphate as the product. Now, why is that enzyme so important for boards? There's no pathology. You'd be dead if that was a complete enzyme defect. The reason why that enzyme is important, it's been on boards, it's highly regulated. And it's regulated by AMP. So this enzyme is regulated by AMP. Now, let me ask you this before I give you the answer. Do you think, you're a thinking person, don't go memorizing here, do you think high levels of AMP turn on the enzyme or turn off the enzyme? What makes sense to you? High levels of AMP turn on the enzyme or turn it off? That's right, and it turns it on. And why does that make sense to you? Because in hard-working muscle, just picture, in hard-working muscle, when you're exercising, in your muscle cells, ATP is being consumed down to ADP, down to AMP. AMP levels in the muscle cell starts to skyrocket. And AMP is telling, turn on glycolysis because we need more ATP to do muscle contraction. It makes sense. There's no reason to memorize that. It just makes sense. AMP turns on the enzyme. What also makes sense is ATP turns off the enzyme. If there's a lot of you're sitting down right now. You're not exercising. And because you're sitting down, if you just ate a donut, ATP levels are rising. Turn off the enzyme. Why make more ATP? If there's plenty of ATP in the cell, why make more? Turn off the enzyme. So that makes sense. Now, what doesn't make sense, and maybe later on we will make sense out of it, is citrate. Citrate also turns off the enzyme here. So again, this enzyme is highly allosterically regulated. Now let's proceed. We'll come back to this enzyme before I'm done with the page. Now once fructose 1,6-bisphosphate is formed, it sees a low-yield enzyme called aldolase, and aldolase just cuts it right down the middle, splitting the 6 carbons into 2 3-carbon fragments. One of them is dihydroxyacetone phosphate, which is needed to make a glycerol platform for making triglycerides, also involved in electron shuttle system, low yield. But if the dihydroxyacetone phosphate is not needed for that, then there's another low yield enzyme that converts the dihydroxyacetone phosphate to a second molecule of the other 3-carbon fragment, and that's glyceraldehyde 3-phosphate. Since we're making 2 molecules of glyceraldehyde 3-phosphate at this point, from here on, everything's multiplied by 2. Let's proceed. So once glyceraldehyde 3-phosphate is formed, it sees a low-yield enzyme, another low-yield enzyme. But notice the surname, it's a dehydrogenase. And what the dehydrogenase is going to do is pluck out high-energy electrons from the carbon of glyceraldehyde 3-phosphate and puts the high-energy electrons onto NAD to form NADH as the product. The only oxidation step in glycolysis, the only place in glycolysis where high energy electrons are plucked out of carbon. Let's proceed. So it uses inorganic phosphate as well to make the other product called 1,3-bisphosphoglycerate, 1,3-BPG. Now once it's formed, it sees another low yield enzyme, and it turns out that 1,3-BPG is a high energy phosphate in it. And so what the enzyme does, it takes the high energy phosphate from it, puts it on the ADP to form ATP. Because a substrate was used to phosphorylate ADP to form ATP, this is the first of 3 examples of what's known in the body of substrate level phosphorylation. A substrate was used to phosphorylate ADP to form ATP as a product, the first of 3 examples. Now let's proceed. So it generates 3-phosphoglycerate, low yield enzyme, to 2-phosphoglycerate, low yield enzyme, to PEP, phosphoenolpyruvate. And it turns out what you need to know about PEP, it's the highest energy compound in the human cell. It has twice the amount of energy than ATP alone. And PEP is the highest energy molecule in the cell, and because it is, the body uses the third and last enzyme you need to know, called pyruvate kinase, and what it does, it uses the high energy phosphate to phosphorylate ADP to form ATP as the product. Again, catalyzed by the enzyme pyruvate kinase, the third and last enzyme you need to know, making the other product pyruvate, which can then go into the mitochondria to be burned, and we'll talk about that issue later. So this is the second of the 3 substrate level phosphorylation steps, where a substrate was used to make phosphorylate ADP to form ATP. All right, now let me say something here. So that's glycolysis in a nutshell, right there. It wasn't hard, but now a couple items here is how many ATPs net ATP do you get in glycolysis? Net ATP. No, in glycolysis, just glycolysis, no other pathways. And the answer is 2. 2 ATPs is net ATPs. Now here it is, just look at the green arrow here. So where does the 2 come from? -1 at the beginning, -1 by PFK1. +2 right here, because everything is multiplied by 2 here, and +2 there. So a net of 2 ATPs. -1, -1, +2, +2. A net of 2 ATPs because of this issue of the substrate level phosphorylation. All right, now let me say something here as well and show you, just to illustrate here how important the glycolysis is. What I mean by this, you probably think, glycolysis, who cares about glycolysis? Who cares about this kind of stuff, you know? And let me say something here and why you should care about glycolysis. If ever I end up with a heart attack, if I end up with an MI and I collapse and I survive the MI, I'm going to get on my knees. And after thanking God, I'm going to get on my knees and thank glycolysis. Because it's glycolysis is the only pathway that can work anaerobically. Now just think about it. You have an MI, no oxygen is coming to the heart. And the cardiac cells need to stay alive. The cardiac cells, the heart needs to beat. And the cardiac cells need to stay alive. They got to do those membrane potentials and the pumps and so on. Where are the cardiac cells getting their ATP to stay alive? And you're looking at it. Page 172. It's the anaerobic glycolysis. It's the only pathway that can work anaerobically. All the other pathways, fatty acid oxidation, amino acid oxidation, those generate a lot of ATP, but they are generating ATP aerobically. They depend on oxygen to generate ATP. This pathway is the only one that can work anaerobically in the absence of oxygen, generating ATP. And you're going to say, hey, but it only generates 2 ATPs. That's true. But the last I remember, 2 is greater than 0, right? And so those measly 2 ATPs generating glycolysis, those 2 ATPs is it's better than nothing. And that little bit of ATP is keeping the cardiac cells alive, waiting for the oxygen to come back to carry out aerobic oxidation to generate a lot more ATP. You should thank glycolysis, because glycolysis, remember, is the only pathway that can work anaerobically. Give the pathway the respect it deserves. Again, you've got to understand what this pathway is doing for me. And you, under ischemic conditions, is doing everything it can to generate ATP to keep alive. Keep in mind. All right, now, this enzyme, pyruvate kinase, why is it important for you to know? And the reason is it is regulated. Some books like First Aid talk about the regulation of the enzyme. I have never heard about the regulation being on boards. Forget about it. Don't worry about the regulation of pyruvate kinase. Because why? Because what they're going to, if they ask you about pyruvate kinase, it's in the context of pathology. Because there is a well-known pathology associated with PK deficiency, pyruvate kinase deficiency, and look at the clinical manifestations. They have hemolytic anemia, which turns out to be the second most common cause of hemolytic anemia in humans, is a PK deficiency. In addition, they have increased BPG, that's increased 2,3-BPG. That should be ringing bells. Increased 2,3-BPG. Oh, physiology. Ooh. And they have no Heinz bodies. All right, so now we have explaining to do. You can memorize all these clinical manifestations, but if you think about it, they make sense if you understand glycolysis. All right, now here comes the issue here is, how do you explain the hemolytic anemia in PK deficiency? Now, the way you interpret this is, I could give you the answer right at the beginning, but I'd rather have you think about this. Now, think about it. Now, this harkens back to I guess histology or some place where we talked about the differentiation of the nucleated red cell. Remember when we talked about the differentiation of a nucleated red cell, how the nucleated red cell differentiated all the way down to the reticulocyte, down to the erythrocyte. And all during that differentiation, the red cell is throwing out the nucleus, the ER, the Golgi, the peroxisomes, all the organelles are being thrown out. And by the time that red cell became a full-blown erythrocyte, just 2 metabolic pathways remain. And they are glycolysis and the hexose monophosphate shunt. So now we have to ask, well, why didn't the erythrocyte throw out those pathways? And you say, well, and the reason is because they need the pathways. So then we've got to ask, why does the erythrocyte keep glycolysis and why does it keep the hexose monophosphate shunt? And if you understand it, you understand the pathology. All right, now here comes the glycolysis. So in glycolysis, we must ask, why does it need the ATP for and what's it need the NADH for? Now, if you understand why it needs ATP, why does the erythrocyte need ATP? It explains the hemolytic anemia. Let me see your answers here. Why do you think, so the erythrocyte keeps glycolysis because it needs ATP. But what's the need the ATP for? And you understand the hemolytic anemia. Let me see your answers. What does it need the ATP for? Give everybody a chance to type. What's the need the ATP for? Not to carry oxygen. Remember, no, not to carry oxygen. Not from the. And the answer, there we go. Now, Daniel, that's right, and Mithra and Perna, that's right. And the answer is, it's all those cation pumps, the sodium, potassium ATPase, the calcium pumps, and so on. Every cell has those pumps, and they need their active transport systems. And they need ATP to pump out those salts. And where's the ATP coming from in the red blood cell? It's coming from glycolysis here. And if the glycolysis is defective in PK deficiency, they cannot pump out the salts, and therefore the membrane integrity is impaired. The cells lyse. It makes sense. Now let me ask you this. If that enzyme is defective in the red blood cell causing the hemolytic anemia, what about the other cells in the body, like the brain cells? The enzyme is going to be defective in the brain, the enzyme is going to be defective in the liver, but why isn't the liver becoming damaged? Why isn't the brain lysing? Why are the other cells fine? Why is this disease manifest in the red blood cell? There we go, because you're a thinker. You're a thinker. You say, well, that's because the other cells have other ways to make ATP, like fatty acid oxidation, amino acid oxidation, whereas the red blood cell has just two pathways, and one of them makes the ATP, and without that ATP, that's why the red blood cell suffers. Look how it makes sense, right? And notice here, of the two pathways that red blood cells have, both of them start with glucose. That now we have come full circle. This is why the body goes all out to maintain blood glucose, because of the erythrocyte. And then if you ask, what does the erythrocyte need the glucose for? Because it has two pathways, glycolysis and the hexose monophosphate shunt, and both start with glucose. Not mannose, not galactose, start with glucose. Just makes sense. Coming full circle. Why the body goes all out to maintain blood glucose here. All right, now the next item here. This one's a harder one to figure out. Now, if you looked at the videos, please don't respond, because I talked about it in the videos as well. So please don't respond if you have it in front of you there. You wrote down the answer. But now, why does the erythrocyte keep the NADH for? What does it need the NADH for? It needs ATP for the cation pumps. But what's the need the NADH for? Now, this answer is a hard one to figure out. Why does the erythrocyte need NADH? To get rid of free radicals and so on. Good try, Hilal. No, that's NADPH. That's the purpose of the hexose monophosphate shunt. The issue of the antioxidants. That's NADPH. Not a buffer. No, not good try. Good try. Not catalase. No, that's NADPH. What's it need? Electron transport. Urege, Mithra. No, there's no mitochondria. There is no electron transport chain in the red blood cell, right? Remember, it's just a bag of hemoglobin in the interior. There are no organelles in the red blood cell. So nobody yet. What's it need? At least you're trying, Urege and Mithra, but not the electron transport chain. Here comes the answer. It has everything to do with something called methemoglobin. What does that mean? What's the difference between methemoglobin and hemoglobin? Now again, methemoglobin cannot carry oxygen. That's not what I'm asking. But what is the difference between hemoglobin and methemoglobin? What's the difference between hemoglobin and methemoglobin? And the answer is one electron. One lousy electron. In hemoglobin, iron is in the +2 state. In methemoglobin, iron is in the +3 state. And as we'll talk about tomorrow, in me and you every day, A little bit of your hemoglobin, up to 3% of your hemoglobin every day converts to methemoglobin by accident. You lose an electron from the hemoglobin and it converts to methemoglobin by accident. Up to 3% every day of your hemoglobin does that by accident. Now, tomorrow we'll talk about why. But the idea here is, if that's all there was, after a week to 10 days, all of your hemoglobin would be methemoglobin, you couldn't carry oxygen anymore, and you would be dead. The body says, you know, I want to live. And if you want to live, you got to do something about that. And the way the body deals with this is, there is an enzyme in the red blood cell that takes the electron from NADH, which comes from glucose, and takes the electron and purposely puts it back into iron plus 3 to restore iron plus 2 in the hemoglobin, so that you and I can carry oxygen here. There it is right there. It just makes sense here. And so in the methemoglobin converting to hemoglobin every day. Now keep in mind, because tomorrow I'm going to ask you about the pathology just of that. There are people in this world who cannot convert methemoglobin to hemoglobin very well. And as a result, their methemoglobins are very, very high in the blood, and that disease is called methemoglobinemia. And as a result, what do those people look like, by the way? What do those people look like who have methemoglobinemia? What do they look like? That's right, they look cyanotic. They look blue. That's right, the blue people, because our methemoglobin levels are elevated here. Now, not now, but tomorrow. Don't answer right now. But tomorrow, I'm going to ask you, I want you to think about it tonight. What's the name of the pharmacology used to treat methemoglobinemia? And then what's the name of the drug? And then I'm going to ask you tomorrow, why do you give that drug? What's it doing? But don't now. I'm just trying to get you to think. The integration, the biochemistry with the pharm, with the pathology. Okay, so that explains the hemolytic anemia here. Okay, now in addition, they have increased BPG. Again, that's 2,3-BPG. And what's happening here is, when this arrow is not working because the enzyme is defective, the substrate accumulates. You're going to hear this over and over again in metabolism. When one enzyme is not working, the substrate accumulates and starts to back up. And it backs up to 2-phosphoglycerate, backs up to 3-phosphoglycerate, backs up to 1,3-BPG. And in red blood cells, the 1,3-BPG converts to 2,3-BPG. And as you know, the 2,3-B, the excess monophosphate shunt, that's the NADPH. But now the idea of the 2,3-BPG, as you know, that has a lot to do with oxygen affinity for hemoglobin, as well as oxygen unloading, the direct integration with physiology. Now let me ask you this. With oxygen affinity for hemoglobin, when BPG starts to rise, where should the arrow be? Increase, decrease oxygen affinity for hemoglobin, or no change? Let me see your answer. Increase, decrease, or no change when BPG rises. That's right, it decreases oxygen affinity for hemoglobin, meaning increase oxygen unloading. In other words, these people have a hard time carrying oxygen around because it gets unloaded much too quickly. So they have a lot of peripheral issues, peripheral tissue issues, because the oxygen gets unloaded much too quickly. Now let me say something about the oxygen saturation curve. If you never understood the oxygen saturation curve, the significance of it, yes, I know Dr. Wilson talked about it. Yes, I know you heard about it in first-year physiology, another course, and so on. If you never understood the oxygen saturation curve and its significance, give me a chance, because that's the next figure. But right now we're on this figure. And next figure is the biochemical point of view of the all-important oxygen saturation curve and the issue of the BPG. Why do you need the BPG? Coming up on the next figure after the break. But now let's finish off this figure. Okay, so they have increased BPG and they have no Heinz bodies as well. And because they have no Heinz bodies, we'll talk about that later, in the number one most common cause of hemolytic anemia, as everybody knows, that's G6PD deficiency here. Okay, so again, that's this major disease, PK deficiency. Notice the integration of this biochemistry and explains the pathology of the hemolytic anemia here. All right, now let's come back to the normal situation. The normal situation. No pathology here. Now suppose you eat a high carbohydrate meal. When you eat a high carbohydrate meal, you're going to run glycolysis. So if you eat a donut coming up at the break soon, you run glycolysis, the pyruvate goes in the mitochondria, you generate a lot more ATP. So you can imagine soon after eating a high carbohydrate meal, the blood, what it's going to happen is ATP levels in the cell will start to rise. Does that ring a bell? Does that ring a bell? Ding, 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 ding. Ah, the ATP in turn inhibits PFK1. And by inhibiting PFK1 in all the cells in the body, glycolysis comes to a grinding halt. So in other words, soon after eating a donut, if you're sitting down, you're not consuming that ATP in large amounts, ATP levels rise in all the cells, and glycolysis eventually shut down. And if that's all there was, you and I would never be able to make a fatty acid. Never make a fatty acid. Why? Because to make a fatty acid, down here at the bottom of the figure in mitochondria, to make a fatty acid, you need to get by this arrow. You need to convert glucose down to pyruvate. And if PFK1 is blocked by the high ATP, you would never make a fatty acid because ATP inhibits PFK1. And therefore, you would never make pyruvate in a high carbohydrate meal. You would never make a fatty acid. But as you know, humans can make fatty acids, so there's got to be something special. Here comes the phrase, in the liver and liver only, no exceptions. What happens is there is an enzyme called PFK2, not PFK1. This is PFK2, phosphofructokinase 2. And what does the enzyme do? It just takes out a little bit of the fructose 6-phosphate, it diverts it out of glycolysis, and this enzyme, because it's a kinase, uses ATP to phosphorylate carbon 2, making fructose 2,6-bisphosphate as the product. Now the so what is, the fructose 2,6-bisphosphate overrides the ATP citrate inhibition. And by overriding the ATP citrate inhibition, you then, PFK1 will now work, and then driving glycolysis to make pyruvate, to make fatty acids in the liver. So in other words, when PFK2 is active, it makes that product, which makes PFK1 active. Whereas in all the other cells in the body, PFK1 shuts down. Location, location, location. Cannot emphasize enough. In all the other cells in the human body, after a high carbohydrate meal, glycolysis is shutting down. But in the liver, it's working overtime because PFK1 is now making a product which makes PFK2 makes a product which makes PFK1 active. Now you might imagine PFK2 is what turns on PFK2 in eating a high carbohydrate meal. You gotta ask, well, what's turning on PFK2 in the liver? And here it is. It's gotta be insulin. Insulin turns it on by dephosphorylation mechanisms. As you might imagine, glucagon turns it off by phosphorylation mechanisms. But the idea is that when you have insulin around in a high carb meal, it turns on this enzyme, which then turns on PFK1, driving glycolysis. Okay, everybody with me here? Everybody with me? That's right, insulin's the boss. Because if you understand this, okay, now this is where you need to think. You need to think. Because if you think, that's big, what's the big deal here? He's boring me. Here comes the issue of pathology. Look at the top of the figure. Look at the top of the figure. And now tell me, what's going to happen in diabetes in a liver of a diabetic. What's going to happen here? Look at the top of the figure, explains the pathology. Look. So notice here, in type 1 or type 2 diabetes, when you don't have the insulin effect in the liver, then as a result, what happens to PFK2? It's never turned on. And if it's never turned on, what happens to PFK1 in the liver? Well, the liver becomes like every other cell in the body. When there's no PFK2, then the PFK1 was shut down because of all the ATP around. It was shut down, and now the liver is like every cell in the body. In me and you, the liver is working overtime in glycolysis, and the other cells are not doing glycolysis. But now in a diabetic, all the cells in the body, including the liver, glycolysis stops because of all that ATP. Now notice what's going to happen with this. So when PFK1 shuts down because of all that ATP in the liver, we're talking about the liver now, then fructose 6-phosphate starts to accumulate. And fructose 6-phosphate backs up to glucose 6-phosphate. And when glucose 6-phosphate accumulates, what does it do? It shuts down hexokinase. So again, when PFK1 shuts down, its substrate fructose 6 accumulates. It backs up to glucose 6-phosphate. It accumulates. And by doing so, inhibits hexokinase. So you can imagine now, a diabetic eats a chocolate bar or a donut, the glucose, excess glucose now coming into the liver, but hexokinase is shutting down. And so the glucose comes into the diabetic liver and can't be trapped by hexokinase. And you're going to say, well, don't worry because the liver has glucokinase, the main enzyme for trapping the glucose. But now, we notice over here how insulin turns on glucokinase. And in diabetes, there's no insulin effect. So what happens to glucokinase? There ain't none. No glucokinase. So now, let me see your answers here. Let me see your answers. What is the overall effect now? Because of all the stuff happening at the top of the page, no insulin effect. What is the net effect in the liver? That's right. That's right. No glucose trapping. So again, in other words, it's so excited because the biochemistry is explaining something here, and I want students to understand this. You can imagine now, a diabetic eats a chocolate bar, a donut, and in all cells in the body, glycolysis is stopping because of all the ATP, and all the excess glucose is supposed to go to the liver, and insulin is supposed to turn on glycolysis to drive the glucose to making fatty acids. But now in a diabetic situation, there's no insulin effect. All this excess glucose comes into the liver, but can't be trapped. So what happens to the glucose in the liver? It goes back into the blood. Because it can't, no phosphate on the glucose goes back into the bloodstream, circulates and circulates and circulates, looking for a home. But there's no room in the inn. And glucose is looking to get inside cells, but can't get inside cells because glycolysis is shutting down in 10 trillion cells. So what happens in the glucose in the blood? It stays in the blood in hyperglycemic conditions, and slowly but surely, glucose latches onto the hemoglobin A1C, latches onto the transporters of the kidney, the renal problems, latches onto proteins like collagen, causing the blood flow impairment and so on. Gets into the eye. We'll talk about retinopathies a little bit later once we get to galactosemia. But the idea is the glucose in the blood is causing havoc to the vasculature. Tremendous idea here. It makes sense. And you thought glycolysis wasn't important. Oh man, yeah, you thought glycolysis, who cares? Oh yeah, Glycolysis is so important, and we're not done with glycolysis. I'm telling you right, we're not. What we're going to do is we're going to take a 10-minute break and come back and get into some physiology. We're not done with, don't turn the page yet. We're not done with the page. So let's take a 10-minute break, and then we'll be back, and I'll see you in 10. But you got to come back. A lot more physiology coming up. Okay, see you shortly. Okay, we're going to finish off this figure on glycolysis before Natalie falls asleep. I know she says she's fallen asleep with this stuff. What can I say? I feel bad for you, Natalie. Doing the best I can, but this stuff is, what do you want me to do? This is where you got to toughen up. You know, if you want to understand it, you're paying a lot of money to be here, and hopefully you can stay awake for this last part. I know. I'm trying to, like Daniel said, you got to suck it up. I know. We just got to do what you can. All right, now this next part here is a direct integration of biochemistry with physiology. Now this has to do, on this figure right here, it has to do with this step right here, this NAD to NADH. Now it turns out, and this is what's critical to understand, the amount of NAD you have in a cell is tiny. You don't have very much NAD in the cell. And so therefore, when you consume it to run glycolysis and you generate NADH, to keep doing glycolysis, you have to recycle the NADH back to the NAD. And thereby, when you have the NAD, then you keep doing glycolysis. So then the question is, well, how do you recycle the NADH back to the NAD? And the answer is, in other words, what do you do with the electron in NADH to recycle it back to NAD? And the answer is, it entirely depends on the presence or absence of oxygen. If you can breathe in enough oxygen, then the electron from NADH is given to the oxygen in the electron transport chain in the mitochondria, and by doing so, you make a lot more ATP. And the more ATP you make, the faster you can run, the higher you can jump, the longer you can swim. And so if you're a conditioned athlete, what you want to do, you're trying to condition your lungs to breathe in more oxygen per unit breath, and the more oxygen you breathe in per unit breath or per unit time, the more ATP you make. But on the other hand, if you cannot breathe in enough oxygen, if you're a smoker, for example, or you don't exercise, then your lungs are not conditioned to breathe in very much oxygen. And if you do not breathe in very much oxygen, then as a result, to recycle the NADH in the exercise back to the NAD to keep doing glycolysis, then there is an enzyme called lactate dehydrogenase in the muscle cells, in hard-working muscle. And what it's going to do is take the electron from NADH and put it purposely onto pyruvate, the end product of glycolysis, to make lactic acid as the product. And then the lactic acid in the muscle cells then will start slowing you down. You get muscle cramping, and it gets spilled in the blood. The pH of the blood starts to drop, and you start slowing down. So the idea of exercise, and why you should like to exercise, because the more oxygen you breathe in, then the more ATP you make, because you're doing aerobic metabolism. But if you cannot breathe in very much oxygen, like for example, if a smoker runs up a flight of steps, by the time they get to the top of the steps, they're going like this. I'm so tired. That's because they can't breathe in enough oxygen. And because they don't, to recycle NADH back to NAD, to keep doing glycolysis, to make a little bit of ATP, to do muscle contraction, LDH enzyme is taking an electron and puts it on the pyruvate to form lactic acid. And that slows you down and become very, very tired. Let me stress, this is why you should like to do exercise, why you should like to do aerobics and things like that, because you're conditioning the lungs to breathe in more oxygen. But let me stress, no human being can breathe in enough oxygen to meet the demands of glycolysis in hard-working muscle. No human being. Even the fastest human being on earth, Usain Bolt, the Jamaican, the guy who won the Olympics, the 100-meter dash, That guy's an amazing athlete. I love watching that guy run. He's an amazing athlete. But even him, he could not breathe in enough oxygen to meet the demands of glycolysis. How much glycolysis is going on in the human body when you're exercising? Not even him can breathe in enough oxygen. What I mean by that is when he runs a 100-meter dash in 9.5 seconds, he cannot run a 200-meter dash or a mile or a marathon at that same pace because he cannot breathe in enough oxygen to meet the demands of glycolysis. So as a result, he cannot breathe in enough oxygen. Nobody can. And then he starts generating a lot of lactic acid, and then he starts slowing down. That's why he cannot run, nobody can run a marathon at the same clip, same pace they run a 100-meter dash. How much glycolysis is going on in hard-working muscle. It's amazing. That's right. It is amazing how much glycolysis is going on. Now, what I want to do here is we're done with this figure finally. But what I want to do is to talk about the biochemistry of the oxygen saturation curve. This business of why you must deliver oxygen to the cells here. Now what you got here, again, if you never understood this in physiology, this is the biochemistry point of view. Now remember, hemoglobin has 2 alpha and 2 beta subunits. And now what I want to do is, I want to talk about, you see 2 curves. Here's hemoglobin A, and here's hemoglobin A plus BPG in the red blood cell. Now what I first want to do is explain what would happen if you did not have BPG in the red blood cell. You just had hemoglobin in the red blood cell. So to explain what goes on, let's get rid of that curve and just talk about what would happen if you had erythrocytes with hemoglobin A in the interior. Now remember from physiology, this is the oxygen tension in the lung area, about 100 millimeters of mercury. And here's the oxygen tension in the peripheral tissues, about 30 or 40 millimeters of mercury. Now what does the curve say? The curve says is when those red blood cells with hemoglobin in the interior get pumped to the lungs, the oxygen tension of the atmosphere is very high, and the oxygen tension, 100 millimeters of mercury, will now force the oxygen onto every subunit of every hemoglobin, of every erythrocytes. In the lung area, the oxygen tension 100 millimeters of mercury, and what that means is every subunit of every hemoglobin, of every erythrocyte has the cargo of oxygen. The hemoglobin is 100% saturated. Exactly what you want to happen in the lung area. The hemoglobin has the cargo of oxygen. Again, exactly what you want to happen. But then when those red blood cells leave the lung area, as they start to travel through the arteries, the oxygen tension is dropping from 100 millimeters of mercury down to 80, down to 60, down to 40, and by the time those erythrocytes get to the tip of my finger, at the tip of my finger, you probably never thought about this, but at the tip of that finger right here, there's an epithelial cell. There's a muscle cell over here that needs oxygen to suck up electrons in metabolism. So now just picture at the tip of my finger right here, there's a red blood cell that's now has the cargo of oxygen. Here's the bloodstream in the middle, and here's an epithelial cell or muscle cell that needs the oxygen. And so the red blood cell has the cargo. But now the oxygen tension is very low, about 30 millimeters of mercury. But what does the curve say? The curve says this, even though the oxygen tension is very low, it's still high enough to keep the oxygen forced on the hemoglobin A, because hemoglobin A is still 100% saturated. It still has the cargo of oxygen. The oxygen tension is very low, but cannot come off hemoglobin A, because as it wants to come off, the oxygen tension is very low, but still high enough that it forces the oxygen on hemoglobin A. It would never come off hemoglobin A at the tip of my finger. So the epithelial cell never gets the cargo of oxygen. The red blood cell has the cargo, but it's not delivered. And then what happens? Then those red blood cells leave my fingers, they travel to the tip of my toe. At the tip of my toe, there's an epithelial cell down there. There's a muscle cell down there that needs the oxygen to suck up electrons in metabolism. And so when the red blood cell brings the oxygen cargo, the oxygen tension at the tip of my toes is about 25 millimeters of mercury, very, very low. But notice the curve says, even though the oxygen tension is very low, it's still high enough to keep the oxygen on hemoglobin A. It just forces it on hemoglobin A, prevents it from coming off, and then never be delivered. And then those red blood cells leave my toes, they circulate and circulate with the cargo, and it's never, ever delivered. There's two things that must happen to the oxygen. You got to pick it up by the red blood cell, and you have to deliver it. It's two different things that must occur. So the picking up the cargo is fine. The delivery is the problem. It never comes off hemoglobin A, even if it's a low oxygen tension in the peripheral tissues. And you'd be dead if that's all there was. All right, now here comes the other issue. So now this is why you need the BPG, because BPG binds to the beta subunits. Now remember, when the oxygen binds to the hemoglobin, it's a cooperative effect. The first oxygen binds and then enables the second oxygen to bind easier, the third oxygen goes on even easier, and the fourth oxygen goes on the easiest. It's a cooperative effect going on and a cooperative effect coming off. Now what you need to know is the BPG binds to the beta subunits here. All right, now let's bring back the other curve here. So bring back the other curve. Now here is the red blood cell with hemoglobin in the interior, and also in the interior, the real life situation, you have BPG in the interior. Now what does the curve say? This curve right here, what does it say? What it says is when those red blood cells with hemoglobin A in the interior along with BPG, when they get to the lungs, and the oxygen tension is 100 millimeters of mercury, very high, and it forces the oxygen onto all four subunits of every hemoglobin of every red blood cell. Even though there's BPG in the interior of the red blood cell, the oxygen is now put onto all four subunits of the hemoglobin. Exactly what you want to happen. In the lung area, it's 100% saturated. Every subunit of every hemoglobin of every erythrocyte has the cargo. Perfect. Exactly what you want to happen. But now as those erythrocytes leave the lung areas, as they leave the lung area, what's going to happen is the BPG, as the oxygen tension starts to drop and drop, BPG starts to bind to the beta subunits and starts kicking the oxygen off the hemoglobin A beta subunits. They come off the beta subunits and then they come off the alpha subunits in a cooperative effect. And by the time those erythrocytes get to the peripheral tissues, again, BPG keeps binding to the beta subunits and kicking the oxygen off. And by the time those erythrocytes get to my peripheral tissues, tip of my toes, for example, how much oxygen remains on the hemoglobin? It's this much remains at 30 millimeters of mercury, and this is how much was there, this is how much remains, and that means this much oxygen was kicked off, was unloaded by the BPG binding to the beta subunits. And that much oxygen comes off and gets into the peripheral tissues to suck up electrons in metabolism. And then what happens? Those red blood cells then go back to the lungs, and the oxygen tension is very high, and then the oxygen tension puts the oxygen back onto the hemoglobin A, and then as they travel through the arteries, BPG kicks it off. The erythrocytes go back to the lungs, pick up the cargo of oxygen, and then BPG kicks it off in the peripheral tissues. go back to the lungs, pick up the cargo of oxygen, and BPG kicks it off in the peripheral tissues under the lower oxygen tension. Beautiful, beautiful how that works. Beautiful. Sorry, Natalie, that we're putting you to sleep, but this is this thing called life. Why you need the BPG. Back and forth it goes. It's incredible why you need the BPG to kick the oxygen off. I'm just pulling your chain there, Natalie. Don't worry, I understand. I get it. All right. So again, the idea, you must understand why you need the BPG, why you need hemoglobin, because you got to pick up the cargo and deliver the cargo. You need the oxygen to carry out aerobic metabolism, which is generating the vast bulk of the ATP in the cell. All right, now what we're going to do is we're going to come back to glucose metabolism later. But what we're first going to do are two other sugars that we need to attend to. And one is galactose, the other is fructose. And why are these two sugars so important? Because the pathways are not essential for life. And so you can have complete enzyme defects, and you're going to see pathology because they're not essential for life. It may not be good life, but people still live with defects in galactose metabolism as well as fructose metabolism. All right, now with galactose metabolism, high yield pathway for the boards. Now let me do the biochemistry first, and then we'll back off to what you need to know for the boards here. Now regarding galactose, where does the human being get galactose? We get galactose by the table sugar called lactose, which is a disaccharide. And then it sees the brush border enzyme called lactase, and lactase then cleaves the disaccharide to the monosaccharides, glucose and galactose as the product. By the way, before we move on here, remember I mentioned to you yesterday that when you go take the National Board exam, because you're going to be stressed out, human nature, you're going to be stressed out and nervous taking boards. And by doing so, you may have a tendency to make silly mistakes. And one way to make silly mistakes is by confusing words which sound very similar. Remember yesterday we talked about the idea that don't confuse the words thymine with the word thymin. Here's another great example of this. I know you know the difference because you're not stressed out. But on board exams, you might confuse the word lactose with the word lactate and the enzyme lactase. They're not the same. They're not the same. Lactose is a disaccharide. Lactate is a 3-carbon end product of anaerobic glycolysis. So again, don't get them confused. They're not the same, lactose and lactase is an enzyme. Okay, let's proceed. Now we'll come back to lactase and pathology in a few minutes. Now the galactose gets absorbed. Dr. Wilson talked about that by the sodium glucose importer issue. But the idea is active transport systems. But once you get galactose in the blood, notice where it goes. Its two major organs are the liver and the brain. If you want to understand pathology, you must understand location, location, location. So galactose is handled by the liver and the brain. We'll come back to the pathology in a few minutes. Now, once it enters the liver and the brain, right at the beginning, there are two enzymes that need to see. And the first enzyme is called galactokinase. And what is the enzyme going to do? It tells you, because it's a kinase, it's going to use ATP to phosphorylate galactose, that's the first word, and thereby forming galactose phosphate as the product. And because the phosphate's on the sugar, it's trapped in the liver and the brain. Everything to do with the pathology. Everything to do with the differential diagnosing of the pathology. You must understand the biochemistry. Once the phosphate's on the sugar, it's trapped. In this case here, the liver and the brain. Let's proceed in the straightforward biochemistry. Then what happens is it sees an even more important enzyme for the boards called the uridyl transferase. Now, what does this enzyme do? What this enzyme does, it needs two substrates. One is galactose phosphate, and the other substrate is a sugar I briefly mentioned earlier in molecular biology, and this is called UDP glucose. Now, what is UDP glucose? It's a glucose donor. Anything in the body right now that has glucose in it, like glycogen, like glucose cerebroside, anything that has glucose in it, the glucose comes from, is donated by UDP glucose. It's a glucose donor, the high energy form of glucose. Now I'm going to abbreviate UDP glucose another way. Here it is right here. This is UDP glucose. It's made up of uracil, the base, ribose, the sugar, and two phosphates. That's UDP. And on the terminal phosphate, there is a glucose. And that's why it's high energy, because glucose is nearby a pyrophosphate. So now what happens is, what does the enzyme do? It takes glucose phosphate off, UDP glucose, and makes it into the product. So it takes glucose phosphate off and replaces it with galactose phosphate. So glucose phosphate goes off, galactose phosphate goes on. And when galactose phosphate goes on, it makes the other product called UDP galactose. And what is UDP galactose? It's a galactose donor. Anything in the body right now that has galactose in it, like N-glycosylation, O-glycosylation, remember your blood groups, A, B, O blood group has galactose in it. Where did the galactose come from? This pathway, by making UDP galactose a galactose donor. There it is. Okay, so there it is. That's the straightforward biochemistry of galactose metabolism. Now, what do you need to know for on the boards? Well, it turns out the number one genetic disorder of humankind, the numbers are staggering. Something like 80 to 90% of Asians are lactose intolerant. Something like 80%, 70 to 80% of Africans are lactose intolerant. 50% of Mexicans are lactose intolerant. A lot of people listening to me tonight, if you haven't already, sometime in your life, you may end up being lactose intolerant. Now, what happens there is the gene for lactase, for reasons unknown, at least unknown to me, the gene for lactase in the mucosa, as a person gets older, the gene goes from euchromatin in the chromosomes into heterochromatin regions. And in heterochromatin, it's never to be turned on again. It gets completely shut down in some individuals. And as a result, there is no gene that's active, and therefore it cannot be made into RNA, made into enzyme, and the brush border will now lack the lactase enzyme. And those people with lactose intolerant, if they drink milk or other dairy products, they become intolerant to lactose, and they end up with diarrhea, bloating, and the cramps. Nothing lethal by any means, but clearly severely discomforting that goes on. Now let me ask you this. How do you explain the diarrhea? How do you explain the diarrhea in lactose intolerance? Severe diarrhea. How do you explain that? That's right, because lactose is highly osmotic. And so so the idea is when lactose comes down the pipe, you cannot absorb a disaccharide. And so therefore, so the disaccharide lactose cannot be absorbed. Out the body it goes. As it goes out the body, it's highly osmotic and draws the water with it. How to explain the bloating and the cramps? How to explain the bloating and the cramps in lactose intolerance? How to explain that? That's right, because in your colon, there are all those bacteria. And those bacteria, like E. coli down there, they're life forms. They want to grow like me and you. They're life forms. Those E. coli in your colon, they're like caged animals down there. But you're not feeding them. Like before you go to bed tonight, you don't say to your bacterial flora, hey, flora down there, have some meat, I'll see you in the morning. No, you don't do any of that kind of stuff. And so you don't feed your bacterial flora. So they're life forms, and they want to grow like me and you. And so now, if you drink lactose in the milk, then what happens is the bacteria see that lactose. They take up the lactose by something called the lac operon. They take up the lactose, they cleave it to the monosaccharides, they go through glycolysis, and now the issue is, what do the bacteria do with the electron in their NADH in their glycolysis? If there's no oxygen in the colon, there's no oxygen in the colon. So how do the bacteria recycle NADH back to NAD? Well, the bacteria take the electron and they put in carbon to make methane, sulfur to make hydrogen sulfide, hydrogen gas, and there it is, anaerobic fermentation. Because there's no oxygen in the colon, they have to do anaerobic fermentation. Okay, now let's proceed. Major disease disorder is the lactose intolerance. Now, in pathology, there are people in this world that can have galactokinase deficiency. There are other people in this world that have uridyltransferase deficiency, and in both cases, they're classified as the galactosemias. Now, notice here, in both cases, they have cataracts, which we'll talk about in a few minutes here. But in galactokinase deficiency, that's all it is cataracts. But in the uridyltransferase deficiency, besides cataracts, vomiting, diarrhea, lethargy, liver damage, hyperbilirubinemia, mental retardation, clearly much more severe. We got explaining to do. You can memorize this, but how do you explain why is one more severe than the other? All right, now the idea here is if the first enzyme is working and the second one is not. If the first enzyme works, the phosphate's put onto the galactose, and notice it's now trapped in the liver and the brain. And if the uridyltransferase is not working, then what happens to the galactose phosphate in the liver and the brain? Well, it's going to accumulate. It cannot exit the liver and the brain. And as a result, everything the liver is supposed to do will not function. So the idea is, and the liver will eventually become cirrhotic. So now don't look at this. Don't look at the clinical manifestations here. Let me see yes or no responses. In uridyltransferase deficiency, do you expect these people to have hypoglycemia? In uridyltransferase deficiency, yes or no? Do you expect them to have hypoglycemia? Yes or no? And the answer is yes. Why? Because the liver is supposed to make glucose, to maintain blood glucose. So if the liver is cirrhotic, they ain't going to make glucose. They end up with hypoglycemia. Do you expect people with uridyltransferase deficiency, do you expect them to have bleeding problems? Bleeding problems, yes or no? Do you expect them to have bleeding problems, yes or no? And the answer is yes. Why? Because the liver is supposed to make all the blood clotting factors. Do you expect people with uridyltransferase deficiency, do you expect them to have jaundice? Yes or no? Do you expect them to have jaundice? Yes. Why? Because the liver has now become cirrhotic, and the liver is supposed to conjugate bilirubin, we'll talk about on Friday. And if now galactose phosphates accumulate in the liver's damaged, the liver cannot conjugate bilirubin, the patients end up with jaundice. Do you expect these people to have mental retardation without looking? Yes or no? You expect mental retardation? Yes. Why? Because galactose phosphates accumulating in the brain. The brain now will start to malfunction, and they'll end up with mental retardation. You expect people with uridyltransferase deficiency? You expect them to have Fanconi's syndrome? Fanconi syndrome in uridyltransferase deficiency? Do you expect them to have Fanconi's? You got to know what Fanconi's means, right? And the answer is no. Why? Because there's nothing wrong with the kidney. So Fanconi syndrome is a kidney problem, and there's nothing wrong with the kidney. Galactose is not metabolized by the kidney. But you will see Fanconi syndrome in fructose metabolism on the next page, because fructose is metabolized by the liver and the kidney, not the liver and the brain. Fructose. You got to know location, location, location if you want a handle on pathology. Here. All right, so again, so that's why it's such a severe disease, uridyltransferase, because it's liver and the brain. Galactose phosphate is accumulating, damaging those organs. Now, the first one, if that's defective, they're not going to have mental retardation or liver cirrhosis. Why? Because galactose comes into the liver. If the kinase is defective, it comes into the liver, says, hello, anybody home? Nobody home? Okay, see you later, and it goes back into the bloodstream. So it's not trapped in the liver and the brain. That's why they don't have the severe symptoms that the uridyltransferase deficiency presents. Okay, now, so that explains why one's more severe than the other. Now, the next item here, and by the way, galactosemias are fairly common, something like 1 in 15,000. So in your lifetime, you are likely to see patients with galactosemias. All right, now another item here. And so how do we explain the cataracts in both cases here? And the idea is that if the galactose kinase is defective, galactose accumulates in the blood. Fairly obvious. If uridyltransferase is deficient, yes, it backs up the galactose phosphate in the liver and the brain, but it also backs up to the galactose. So both of these are accumulating in uridyltransferase deficiency. It's like having a river. And if you have a river and you put a dam in the river, the water backs up to all the tributaries. It backs up into all the tributaries. So it's backing up all the way to the galactose. So in both cases, galactose is accumulating in the galactosemias. And the galactose can come into the lens. And when it comes into the lens, now no one expects you to know the structure of galactose, but it has 6 carbons, 5 of which have a hydrox group. Carbon 1 is an aldehyde. And in the lens there is an enzyme called the aldose reductase. Look at the name of the enzyme. What it does, it reduces the aldehyde to an alcohol, hydroxyl group. And so now carbon 1 will have a hydroxyl group just like the other carbons. And the product, therefore, is called galactitol. Look at the suffix, OL. Every carbon has a hydroxyl group. And galactitol is considered a polyol. Polyol. Every carbon has a hydroxyl group. So what's the problem with galactitol? It's dead end. It cannot be further metabolized, and it cannot exit the lens. The transporters that get galactose into the lens cannot transport galactitol out of the lens. It's now stuck, trapped in the lens. And then as a result, because it's osmotic, it starts to draw water into the lens starts to swell, and that is now the beginning of cataract formation. Now what you're seeing here is a cartoon of a normal lens. Here is the lens in a galactosemic situation, but this is a cartoon. Here is the real life situation, where the lens in fact look opaque. They look cloudy because galactitol is accumulating in the lens, is trapped in the lens, and the cataract can form. So on board exams, if you see that word cataracts, automatically think to yourself, they might be asking me about a galactosemia. Now let me ask you this. What other disease do you want to focus your attention on? If you see the word cataracts, what other disease do you want to focus your attention on? That's right, diabetes. Now how to explain cataracts in diabetics? And it's the same principle as we talked about earlier in glycolysis, where Natalie was falling asleep. In glycolysis, because glucose is accumulating in the blood, because it can't be trapped in cells, because everything is shutting down. Glucose is in the blood. Glucose can also enter the lens, and in the lens it sees the same enzyme, aldose reductase. And aldose reductase takes carbon 1 of the glucose, makes it into a hydroxyl group, and the product now is called glucitol, and also, but more commonly called sorbitol. And sorbitol is osmotic and starts to draw the water into the lens, and that is now the beginning of cataract formation in a diabetic situation. You should ask, why do you have this enzyme in the first place, aldose reductase, if it's causing all those problems with cataracts? And the reason why you have that enzyme is because you generate sorbitol on purpose. Your lens are very viscous on purpose. And so in fact, you have an enzyme which breaks down sorbitol. The problem in a diabetic situation, you generate too much sorbitol, and it starts to accumulate, causing the cataract formation. But the fact that you can metabolize sorbitol, that's why it takes a while to get a cataract in a diabetic situation. But in the galactosemic situation, you can get a cataract right at birth because there's no way to metabolize galactitol. And so there are babies with galactosemies born with a cataract. How fast the cataract forms in a galactosemic situation, because it can't be metabolized at all. But sorbitol can. That's why it takes a while to get a cataract in a diabetic there. All right, so you got it. So you must understand galactose metabolism, high yield. Now the next sugar to talk about here is fructose, and be ready for pathology here. Let me do the biochemistry first. So what we're going to do is two more figures here. We're going to do fructose metabolism and its pathologies. And then after that, we're going to talk about PDH and its pathologies here. And then we'll finish off this chapter here. Okay, so then we're done. Okay, so the idea of fructose, but again, please stay awake here. This is high yield stuff because of the pathology integration. Now, where does a human being get fructose? We get it by eating the table sugar sucrose, which sees the brush border enzyme sucrase, which cleaves it to the monosaccharide glucose and fructose. Where else do you get fructose? You get fructose in the free form by eating fruits and honey. It is the only monosaccharide that you and I get in the free form in appreciable amounts. Whereas glucose, you get it from starch and so on, and same thing with galactose and lactose. But fructose is the only monosaccharide we can get in free form in relatively large amounts by eating fruits and honey. Now the fructose, once it's in the intestine, it gets transported, not by active transport, but I'm sure Dr. Wilson talked about this in GI physiology. But the fructose is now absorbed and comes into the blood, and notice where it goes. Liver and the kidney. Let me highlight that. Liver and the kidney, not the liver and the brain. Everything to do with pathology. Location, location, location. All right, now once it comes into the liver and the kidney, it sees another pair of enzymes, just like galactose did. And the first of these enzymes is called fructokinase. And what it's going to do is put a phosphate onto the fructose, making fructose 1-phosphate as the product. Now let me ask you this, not shown on this figure, what does this enzyme need to work? Let me see your answer. What does this enzyme need to work? Not ADP, not ADP, not ADP. Yeah, that's right. Now we go. That's right. That's right. It's ATP because kinases use ATP. Okay, so they use ATP to put the phosphate onto fructose because the name of the enzyme tells you, fructokinase. How many kinases have we talked about thus far? We talked about protein kinase A, G, and C. We talked about tyrosine kinases. We talked about, tonight we talked about hexokinase, glucokinase, the previous page, galactokinase, and now fructokinases. You got to get them straightened out, and they are easy. Just look at the first word, it tells you what it does. Not hard, no reason to memorize, no reason to panic. It's just a matter of using your mind. Beautiful mind, use it. All right, now once the phosphate's on the fructose, it's trapped in the liver and the kidney. Again, everything to do with the differential diagnosing of the diseases. Now, once it's formed, it sees an even more important enzyme for the board. It's called aldolase B. And what does aldolase B does? It does a hatchet job. It just cuts it right down the middle, generating two fragments, 2 3-carbon fragments, which then go into glycolysis to be burned. All right, there it is. There is the pathology. There is the straightforward biochemistry. Now, if there's an aldolase B, they're going to be an aldolase A. And there is in glycolysis, but there in glycolysis, low yield. Aldolase B is high yield because there is pathology. Now, speaking of pathology, either of these two enzymes can be defective. The genes can be mutated. And you can end up with fructokinase deficiency, which is benign. And by the way, in fructokinase deficiency, in pathology, what is the other name of this disease called? The pathology. What is the other name of the pathology called? You might see it on boards called fructokinase deficiency. What is the other name? There we go. That's right. It's more than just fructosuria, Daniel. More information. And the answer is it's called essential fructosuria. So if you see that phrase, you got to use the word essential here. Essential fructosuria, you say, oh, they're asking me about fructokinase. Here. Now the other one here, aldolase B deficiency. They have lethargy, vomiting, liver damage, hyperbilirubinemia, hypoglycemia, hyperuricemia, the Fanconi syndrome, and clearly much more severe here. And now, what's the name of that disease in pathology called? Aldolase B deficiency also is called what? That's right, it's called hereditary fructose intolerance. So you might see it by that name, either as an option of a vignette or the subject of a vignette. They might say hereditary fructose intolerance and say, well, they're asking me, that's aldolase B, not fructokinase here. All right, now the idea is why is one disease benign and the other is severe? And the reason is if the first enzyme is working and the second one is not, then fructose phosphate's accumulating in the liver and the kidney. And anything the liver is supposed to do, anything the kidney is supposed to do, will now malfunction as time goes on. And you end up with all the jaundice and the hypoglycemia because the liver is malfunctioning, the renal problems as well. They're going to have bleeding problems, yes, and so on. And now some books like First Aid and so on talk about trapping of the phosphate. Yeah, that's true. But the main simple thing is the liver is damaged because something is accumulating here. By the way, you might ask, why is fructose phosphate not going into glycolysis? Because after all, there is a fructose phosphate in glycolysis. But that's fructose 6-phosphate. This is fructose 1-phosphate. The phosphates are in the wrong position. That's why it does not go into glycolysis here. Okay, now why is the other enzyme, when it's defective, why is it benign? Because fructose comes into the liver and the kidney. If it can't be trapped with phosphate, it goes back into the blood. And the only symptomology that might occur in fructokinase deficiency is a little bit of dehydration because the fructose will be spilled into the urine and it's osmotic, just like in diabetics. They'd be osmotic and they might be dehydrated, but other than that, it is a benign syndrome here. Okay, major pathology with the aldolase B, major one. All right, now I got a question for you. A little oral vignette. So, and some babies born under your care. And when the baby is born, you do all these tests and you find out that baby is fine. No problem with the baby is perfect. So, so you give the baby a shot of vitamin K prophylactically and tell the parents, okay, everything is good to go home. Okay, so the baby goes home, the baby's then being breastfed by the mother, and for the first 5 or 6 months, everything is going fine with the baby. The baby is developing perfectly. But then after about 5 or 6 months, when the baby starts to become weaned, the baby's now being fed all those Gerber jar things, you know, those Gerber peaches and pears and whatever. And then the parents start to notice that the baby's becoming lethargic, the baby's becoming jaundiced, and then vomiting, and then the parents rush the baby back to you as a pediatrician. You examine the baby and you find out indeed the baby is jaundiced, the baby has a sugar in the urine, you examine the sugar, you find out it's fructose, and you tell the parents, well, unfortunately, your baby has hereditary fructose intolerance. So here comes the question. Why is this a disease that's noticed months after birth when the baby is weaned? Yet in the first few months, the baby is perfect. Why? Why? Not the enzyme in breast milk, no, not the enzyme. It's the sugar in breast milk. The sugar in breast milk is lactose, and the baby has nothing wrong with galactose metabolism. The baby can handle galactose perfectly. But then when the baby's weaned off the milk, no more lactose, and now the baby's being fed those peaches and pies and cookies and chocolates and donuts and things like that, and in those foods are sucrose as well as fruits and honey with all that peaches and pears and so on, all that fructose. And then if the baby has hereditary fructose intolerance, they can't handle that fructose. That's when the disease manifests. And then you might imagine, then for the rest of their life, you would tell the child or the parents of the child the baby cannot have any type of processed sugars with the sucrose in there, because then the baby will be fine. So that for the rest of their life, they just got to avoid sucrose. And you might say, man, that's pretty bad on the child. That's true, but it beats liver damage, right? You know, so the idea is they just have to avoid. There to be a dietary control for the rest of their life. All right, now the question now becomes, in a fructosuria, do you expect to see the baby having a cataracts? Let me see your answer. Do you expect to see cataracts in a fructosuria? And the answer is no. Why not? You see cataracts in galactosemias, but not fructosurias. Why not? Why not? Has nothing to do with trapping. There we go. Rouge. That's right. There it is. Is because Maritza. That's right. And because fructose is a ketose, not an aldose. This is sometimes why structure is sometimes important to know. Galactose has an aldehyde group. Fructose does not. Fructose has a ketose, not an aldose. And so it goes into the lens, but aldose reductase says, hey, who are you? I don't recognize you. And so fructose goes back out because fructose does not have an aldehyde group. No cataracts in a fructosuria. So that's a big clue on national board exam. If they talk about a sugar in the urine of an individual, it's not a diabetic situation. Well, how do you know it's a galactosemia or fructosuria? You look for the word cataracts. If they do, it's a galactosemia. If they don't, it's a fructose problem. Major item here. All right, one last figure, won't take very long, and then we'll finish this chapter. And that's this all-important enzyme called pyruvate dehydrogenase. Major enzyme for the boards. Last figure, then we're done. Won't take long. But please bear with me on this all-important enzyme. Now, what does this enzyme do? We're coming back to glucose metabolism. Now, what happens is when glucose enters the cell, it goes through glycolysis in the cytoplasm of all cells in the body. But then the product is pyruvate. If there are mitochondria in the cell, obviously not red blood cells, but all the other cells, yes. Then pyruvate sees this all-important enzyme called pyruvate dehydrogenase. Look at the surname, dehydrogenase. What it's going to do is pluck out high-energy electrons from the carbon of the pyruvate, puts them on the NAD to form NADH. And then the NADH then goes into the electron transport chain. We'll talk about tomorrow, first thing. And then you generate ATP from the NADH aerobically. This enzyme also splits off CO2, and the two remaining carbons of the pyruvate are put on the coenzyme A, making acetyl-CoA as the product. In terms of the last straightforward biochemistry, the acetyl-CoA inhibits PDH. All right, now the idea of so this enzyme is regulated allosterically, inhibited by acetyl-CoA. Now, why? Now, this enzyme, in terms of the mechanism, it is well known. Now, depending on where you went to medical school, your instructor may have spent a whole hour of your life talking about the mechanism of this enzyme. It's a very complicated mechanism, but it's for the boards, useless information. Don't worry about the mechanism of the enzyme. They ain't going to ask you about the mechanism of the enzyme. What's important to know, though, is what does the enzyme need to work to carry out this mechanism? And it needs 5 different coenzymes. Thymine pyrophosphate coming from the vitamin thiamine, lipoic acid, which does not come from a vitamin, your body just makes it. Coenzyme A coming from pantothenic acid, FAD coming from riboflavin, NAD coming from niacin. Now there's an easy, now they might ask you what vitamin is used to make coenzymes this enzyme needs, PDH, or they might ask which of the following coenzymes this enzyme needs. Now there's an easy way to remember what's the 5 coenzymes this enzyme needs to work. And all you got to do is look at the first letters. Now normally I don't like mnemonics, but here's a great example of a mnemonic which is very useful. And that is the mnemonic, tender, loving care for Nancy, where the first letter tells you helps you know what's the coenzyme the enzyme needs to work. Tender, loving care for Nancy. Now, if you're not going with a girl named Nancy, you would say tender, loving care for no one. Okay, so again, there are 5 different coenzymes this enzyme needs to work. Now, this will be important because there'll be 2 other enzymes that need 2 other enzymes that have the same mechanism, and you can recognize them because they generate NADH, they generate CO2, and most importantly, they make a coenzyme A of a product. And they are all tender, loving care for Nancy enzymes here. All right, that's another reason why you need to know the enzyme, what it needs to work. And the very last thing is this. You must know that one of the vitamins needed to make coenzymes is thiamine. And you must know that thiamine is deficient in alcoholics. And the reason is because there's very little thiamine in alcoholic beverages. There's no thiamine in tequila, for example. And alcohol inhibits thiamine absorption. So you must know that alcoholics are deficient in thiamine. All right, now then I'm going to conclude here by giving you a quick oral vignette. Here comes the question. Suppose on board exams they say to you that you are in your clinics and some long-term alcoholic comes into your clinic. And you know a lot of long-term alcoholics tend to be very thin, they haven't eaten very well, so you want to be a good doctor. So what do is you IV a solution of glucose into them, and shortly thereafter the patient dies. You killed that patient by IVing a solution of glucose into them. Here comes the national board with a question. What did the patient die of? Let me see your answer. Again, one more time. A long-term alcoholic comes into your clinic. You want to be a good person, to IV a solution of glucose into them. The patient dies in your hands. What did the patient die of? That's right. Danielle, that's right. Takir, Andrea, that's right. Well done. Samantha, that's right. And the answer is lactic acidosis. This is how the National Board asks you about metabolic pathways without asking you precise details. Globally, you've got to know the metabolic pathways. So how do you explain the lactic acidosis? It's simple. You got to know the fact that alcoholics are deficient in thiamine. And any enzyme that requires thiamine will not work. And the first enzyme in metabolism that requires thiamine is PDH. So here it is at the top. If you IV this solution of glucose, it goes down through glycolysis down to pyruvate. No problem. And then if there's no thiamine around in alcoholics, PDH will not work. I keep telling you, when one arrow doesn't work, in this case here, nutritionally, not genetically, nutritionally. So if one arrow does not work, the substrate accumulates. And when the substrate accumulates, it spills into other metabolic pathways. What other metabolic pathways can pyruvate go into? LDH converts it into lactic acid. And lactic acid is dead end. It builds up in the liver, and goes into the bloodstream, and it lowers the pH, and you can die. There it is, lactic acidosis, caused by a situation in alcoholics. There it is. It makes sense. All right, now what should you do as a physician with a long-term alcoholic? What should you do? What should you do with an alcoholic? And they come into your clinic. What should you do? What do they tell you in pathology you should do with an alcoholic? That's right. You first give them thiamine. That's right. You first give them thiamine, and then you IV the glucose, and then PDH will work, and everything will be fine. There you go. There it is. Just makes sense. You give them some thiamine, and then you IV the glucose, and everything's hunky-dory. They'll be fine. Okay, everybody, we're done for the day. So we're done now with this chapter, chapter 12. I think, and then so tomorrow in the first hour, we're going to do, we're going to skip over the Krebs cycle, because that's going to be low yield for the boards. It's essential for life, but for the boards, low yield. So the first pathway we're going to get into tomorrow is the electron transport chain in the next chapter. And then after that, we'll get into glycogen, alcohol metabolism. We're going to get into the hexose monophosphate shunt, major for the boards. So we'll do all that carbohydrate stuff in the first few hours tomorrow. And then at the end, we'll start to get into cholesterol stuff in the following chapter. So that's what's on tap for tomorrow. High yield stuff coming your way. So we're done for the day. I'm going to post a few questions on the Facebook. Thank you so much for showing up tonight. You guys have been very attentive. Excellent job of paying attention here. Thank you all so much. Have a great evening, and I'm looking forward to seeing you again tomorrow night. Take care, everybody. God bless you all. See you soon.